Somewhere inside the cells of a living person, beneath the features visible in a face and beneath the family histories carried in names, there are fragments of a much older story. They do not preserve voices, landscapes, or the names of the dead. They survive as patterns in DNA short stretches or inherited code, whose histories do not fit neatly within the known branches of our species. Some came from Neanderthals. Some came from Denisovan-related populations. Others may have entered the human lineage from groups that have no agreed name, no confirmed skeleton, and perhaps no fossil yet recognized for what it is. The evidence is incomplete, but it has changed the way human origins must now be imagined. The past was not a clean procession in which one species replaced another. It was a world of populations dividing, enduring, meeting again, and sometimes disappearing into the descendants of those they encountered. This is where the idea of Ancestor X begins. It is not the official name of a species, nor the label attached to a single lost individual. It is a storytelling name for one or more ancestral populations inferred from genetic traces that cannot yet be tied securely to a particular fossil group. The scientific reality is stranger. There may have been several such populations. Some lived in Asia and contributed DNA to people whose descendants later reached Oceania. Some may have lived in Africa, where warm climates have destroyed most ancient DNA that might have identified them directly. Others may not have been separate species at all, but long isolated populations within a deeply structured human ancestry. Before we go deeper, take a moment to like the video and subscribe, if this is the kind of history and mystery you enjoy. Because this mystery does not begin with a skull rising from the soil. It begins with living people, and with a scientific unease, certain parts of the human genome appear older, more divergent, or more geographically patterned than a simple family tree can comfortably explain. Every human genome is a mosaic assembled through countless generations. Recombination cuts ancestral chromosomes into smaller fragments, each time they pass through a new generation. Old contributions become broken, diluted, or lost. Yet some remain detectable because they differ from surrounding DNA in revealing ways. A segment may carry an unusually deep history. It may cluster more closely with an archaic genome than with other modern human sequences. It may appear frequently in one region but be nearly absent elsewhere. Its length may suggest roughly when it entered a population, while its position on a reconstructed genealogical tree may imply that the lineage behind it diverged long before the known variation among living humans. No scientist looks at one unusual gene and declares that an unknown species has been found. Researchers compare thousands of genomes and test competing models of population history. One model may involve a population splitting and remaining separate. Another may involve later interbreeding. A third may create a similar signal through long-term population structure, where groups remain partly connected but exchange genes unevenly over immense spans of time. Scholars still debate where genuine archaic introgression ends and the shadow of complex population structure begins. It reveals how difficult the question truly is. Human evolution unfolded through populations, not tidy labels. A group could exchange genes occasionally with its neighbours and still remain genetically distinct for hundreds of thousands of years. Another could occupy a distant region and later contribute only a small part of its ancestry to people who survived. By the time geneticists detect such a contribution, the original population may have vanished so completely that its only remaining archive lies in bodies alive today. One of the most dramatic examples emerged from Asia. Then a tiny fragment from a cave in the Altai Mountains yielded DNA unlike anything expected. It belonged neither to a modern human nor to a Neanderthal, yet it came from a population related to both. That population became known as the Denisovans, named after the cave rather than after any ancient identity. At first, they were almost entirely a genetic presence, a lineage known from molecules before science possessed anything close to a complete face. The discovery reversed the usual order of paleoanthropology. Traditionally, bones appeared first and ancestry was argued over afterward. Here, the genome arrived before the anatomy. It revealed that a distinct human population had lived somewhere across Asia, had met both Neanderthals and modern humans, and had left descendants in the genetic sense even though its physical record consisted of only a few fragments. Denisovan DNA was found at its highest levels not around the Siberian cave itself, but among populations far to the southeast, especially in parts of near Oceania. The people carrying these traces had ancestral histories stretching across islands and tropical environments, thousands of kilometers from the cold Altai chamber where the first genome was recovered. This geographic mismatch hinted that the Denisovan story was not the history of one small cave population. It was likely the history of multiple related populations spread across a vast and varied continent. Recent genomic work indicates that ancestors of some communities in near Oceania encountered not one Denisovan source, but at least 3 populations related to Denisovans. These were not necessarily 3 separate species. They may have been long diverged branches within a wider Denisovan world, yet the implication is profound. The single word Denisovan may compress a diversity of human lives almost entirely missing from the fossil record. Some inherited variants appear to have affected gene regulation and immune responses. Others may have helped later populations survive specific environments. But archaic DNA should not be imagined as a collection of ancient superpowers. Much of it was probably neutral. Some was harmful and gradually removed by natural selection. Some became useful only in new contexts. What survives in the record suggests not a deliberate gift from one people to another, but the uneven biological aftermath of meetings whose social details are lost. Beyond Denisovans, even deeper signals have begun to appear. One recent model proposes that the ancestry shared by all modern humans may reflect 2 populations that separated roughly 1.5 million years ago and came together again around the time Homo sapiens was emerging. In that model, the smaller source contributed roughly 1/5 of the ancestry at the moment of reunion. This does not mean every living person carries an intact 20% inherited from a single unknown species. Recombination, natural selection, later population movements, and the assumptions built into the model make the reality far more complicated. Nor does the study identify the smaller population as Homo erectus, Homo heidelbergensis, or any other named fossil group. It reveals a pattern, not a face. The unknown human hidden in our DNA may therefore not be one unknown human at all. It may be a crowd of vanished populations, some archaic, some early members of our own species, some isolated for vast spans of time, and some represented by fossils already found but not yet connected to their genetic legacy. Ancestor X is less a missing box on a family tree than a warning that the tree itself is too simple. Beneath its branches lies a network of separations and reunions, of populations remembered only where their DNA entered another lineage and survived. The search begins by asking how many human worlds have been compressed into the genome of the present. To answer that, the evidence must be followed backward from living chromosomes to ancient fragments, from statistical shadows to caves, teeth, jaws, and landscapes. And in one cold chamber in southern Siberia, a piece of bone smaller than many ordinary archaeological finds would become the doorway into that hidden world. The doorway into that hidden world is not grand. Denisova Cave opens in the limestone of the Altai Mountains, overlooking the Anui River in southern Siberia. Today the surrounding slopes carry forest. But across the Pleistocene the landscape repeatedly changed as cold and warmer phases moved through Inner Asia. Grasslands expanded, forests retreated, animals shifted their ranges, and different human populations entered the region when conditions allowed. The cave offered shelter above the valley floor, a place where smoke, stone flakes, animal bones, and traces of repeated occupation accumulated in layers. For archaeologists, it was already valuable long before anyone understood that it contained evidence for a human population unknown to science. Excavations had recovered tools, animal remains, ornaments, and fragmentary human fossils from sediments disturbed by thousands of years of use. The cave was not a sealed chamber belonging to one people. Neanderthals occupied it. Denisovans occupied it. Modern humans may also have passed through the wider region. Sediment shifted. Objects moved through animal burrows, freezing, thawing, and human activity. Establishing who made a particular tool or wore a particular ornament is therefore difficult. The cave preserves extraordinary evidence, but it does not arrange that evidence into a simple story. In 2008, excavators recovered a small piece of finger bone from one of its layers. The fragment came from the end portion of a little finger and was so modest that, by appearance alone, it could not have transformed the human family tree. It did not display a distinctive brow ridge, jaw shape, or cranial feature. There was no face to reconstruct and almost no anatomy to compare. Traditional fossil analysis could say little more than that it came from a human individual. The bone was initially thought to belong to a young person, though later genetic and developmental work refined aspects of that interpretation. Its importance did not lie in its shape, it lay inside it. Ancient DNA survives unevenly. After death, genetic material breaks into shorter fragments and accumulates chemical damage. Heat, moisture, microbes, and time accelerate its destruction. This is why tropical regions that once supported large human populations often preserve almost no recoverable DNA, while cold caves and permafrost sometimes protect genetic traces for tens or hundreds of thousands of years. Denisova Cave provided unusually favorable conditions. Even there, however, extracting authentic ancient DNA required careful work to distinguish the original molecules from contamination introduced by excavators, laboratory workers, or the surrounding environment. Researchers first examined mitochondrial DNA. The small circular genome passed through the maternal line. Because each cell contains many copies of mitochondrial DNA, it is often easier to recover from ancient remains than the much larger nuclear genome. The expectation was understandable. The fragment would probably belong to a Neanderthal, since Neanderthal remains were already known from the region, or perhaps to an early modern human. Instead, the sequence fell far outside the known mitochondrial variation of both. The result was initially difficult to believe. Contamination, laboratory error, and unusual variation all had to be considered. Yet repeated analysis continued to point toward the same conclusion. The individual represented a maternal lineage that had separated from the lineage leading to modern human mitochondrial DNA far deeper in time than expected for an ordinary member of either known population. It was not enough to name a species, but it was enough to expose a gap in the existing picture. The nuclear genome made the discovery far more powerful. Mitochondrial DNA follows only one narrow line of ancestry, passing from mothers to their children. A population can replace its mitochondrial line through migration or interbreeding without losing most of its wider ancestry. Nuclear DNA, inherited from many ancestors, preserves a more comprehensive record. When researchers reconstructed the nuclear genome from the finger fragment, they found that the individual belonged to a population closely related to Neanderthals but distinct from them. Neanderthals and Denisovans shared a common ancestral population after their lineage separated from the ancestors of modern humans, then divided from each other later. This was no minor branch hidden within known Neanderthal diversity. The genome indicated a population with its own long history. It had diverged, persisted, and encountered other humans. The discovery meant that at least 3 major human lineages had existed close enough in time to interact modern humans, Neanderthals, and the population now called Denisovan. The name came from the cave because there was no ancient name to recover and no established fossil species that could be confidently matched to the genome. A large molar from the cave soon deepened the mystery. It was unusually robust and did not resemble the typical teeth of recent humans. Genetic evidence connected it to the same broader Denisovan population, although not necessarily to the fingerbone individual. Additional fragments followed, but the physical record remained astonishingly small compared with the geographical and genetic importance of the group. For years, an entire population stretching across unknown parts of Asia was represented by a finger fragment, a few teeth, and genetic sequences. The genome also contained evidence that the Denisovan population had not remained isolated. When compared with DNA from living people, parts of the Denisovan genome showed a special relationship with populations in Oceania and parts of Asia. The simplest explanation was interbreeding. Ancestors of some modern populations had met Denisovan-related people, had children with them, and carried portions of their DNA onward. Those fragments survived migrations through Southeast Asia and into near Oceania, even as the Denisovan populations themselves disappeared as recognizable communities. This created a striking contradiction. The physical evidence came from a cold cave in northern Asia, while some of the strongest genetic legacy appeared in people whose ancestors lived far to the southeast. A population known from Siberia had left substantial ancestry in New Guinea and neighbouring regions. Either Denisovan-related groups travelled across enormous distances, or they were already divided into populations occupying much of Asia. The second possibility increasingly fits the evidence, but the full map remains incomplete. Denisova Cave also preserved a more intimate clue. Another small bone fragment, identified through molecular screening rather than obvious anatomy, belonged to a person whose mother was Neanderthal and whose father was Denisovan. This individual would later reveal how directly the populations met. But even the first finger bone had already made such encounters imaginable. Human lineages that looked separate on a diagram were not always separate in life. Their ranges overlapped. Their communities sometimes met. Genetic boundaries remained permeable. There was a temptation to imagine that the discovery instantly solved a mystery. In reality, it created several new ones. Researchers did not know what Denisovans looked like, how they organized their lives, what languages or communication systems they used, or which tools in the cave were theirs. Even their geographic range had to be inferred indirectly. A genome can reveal kinship and population history with extraordinary precision, but it cannot describe clothing, gestures, beliefs, or the meaning of objects left near a hearth. The evidence remains incomplete, and the cave itself complicates interpretation. Some archaeological layers contain material that may have shifted from its original position. Objects associated with symbolic behavior, including ornaments, cannot always be assigned securely to a particular population. Denisovans may have created sophisticated artifacts, but the genome alone cannot prove who shaped every object found nearby. Scientific caution is essential precisely because the discovery is so remarkable. What the finger bone established was not a complete portrait, but a new category of evidence. A human population could be discovered genetically before it was recognized anatomically. The fossil record no longer had the final word on who had existed. DNA could reveal people whose bones has been overlooked, misclassified, or destroyed. It could also expose relationships invisible in anatomy, including interbreeding between groups once imagined as entirely separate. The small fragment from Denisova Cave therefore changed more than one branch of the family tree. It changed the method of searching. After it, unidentified teeth, fragments, cave sediments, and even proteins trapped in ancient material could become evidence for lost populations. The unknown no longer had to wait for a complete skull. Sometimes it could announce itself through a handful of damaged molecules, preserved in darkness until the technology existed to read them. The technology that could read those damaged molecules did not simply reveal a hidden branch of humanity. It taught scientists how to recognize a population that no longer existed as a visible fossil group. Ancient DNA is not recovered as a complete genome waiting inside bone. It arrives in millions of broken pieces, chemically altered, mixed with microbial DNA, and sometimes overwhelmed by traces from the people who handled the specimen. Reconstructing it is less like opening a preserved book than assembling a manuscript that has been shredded, stained, and partly replaced by unrelated pages. The first task is authenticity. Genuine ancient DNA tends to be short, fragmented, and marked by characteristic chemical changes that accumulate after death. Contamination is often longer and less damaged. Researchers compare these patterns, repeat extractions, use clean room procedures, and test whether the sequences behave as expected for old material. Denisova Cave became important not only because it preserved remains, but because cold conditions allowed enough molecular material to endure for genomic reconstruction. Once sequences are recovered, they are aligned against reference genomes. Each fragment is placed where it most likely belongs. While statistical methods account for uncertainty and damage, coverage matters. A low-coverage genome may provide scattered glimpses, while a high-coverage genome allows researchers to identify close relatives, estimate population histories, and detect interbreeding. The Denisovan finger bone yielded an unusually informative genome, making it possible to compare this individual with Neanderthals and living humans at millions of positions. The comparison did not produce one simple percentage. It revealed patterns of shared mutations. If two populations share more derived genetic changes with one another than either shares with a third, that imbalance can point to closer ancestry or later gene flow. Researchers test these relationships across the genome, asking whether the pattern fits a branching tree or whether contact must be added. In the case of Denisovans, a simple tree was not enough. Their genome was related to Neanderthals, yet it also shared distinctive ancestry with some living populations, especially in Oceania. Interbreeding was the clearest explanation. Over generations, recombination broke inherited Denisovan chromosomes into smaller segments. This fragmentation became a kind of clock. Long, archaic segments usually suggest more recent mixture. Shorter segments tend to reflect older events. although selection, demographic change, and repeated contact complicate the estimate. What survives in modern genomes is not a faithful copy of a vanished population. It is a filtered residue shaped by chance, migration, selection, and tens of thousands of years of recombination. Some archaic segments disappeared because they reduced reproductive success. Others drifted away randomly. A smaller number rose in frequency because they became useful under particular conditions. The high-altitude variant associated with oxygen regulation on the Tibetan Plateau is one example of adaptive introgression linked to Denisovan-related ancestry. Yet it is misleading to imagine archaic DNA as a collection of beneficial upgrades. Much of it appears to have been removed, especially near genes where incompatible variants could have disrupted development or fertility. The genome carries both the evidence of contact and the evidence of biological tension afterward. The newer mystery begins where no reference genome exists. Detecting Neanderthal or Denisovan ancestry is comparatively straightforward when a segment can be compared with sequenced archaic individuals. Ancestor X may have left no such reference. Scientists must look instead for the shape of an unknown history. A stretch of DNA may contain unusually old variants, show an exceptionally deep branching pattern, or resist explanation under models containing only known populations. Methods reconstruct local genealogical trees along the genome, following how relationships change from one region to another. This does not allow researchers to see an unknown person directly. It allows them to estimate that an unsampled source may be needed to explain the pattern. Newer methods attempt to detect introgression without requiring a donor genome. They examine tree depth, mutation density, linkage, branch arrangement, and the geographic distribution of segments. Systems such as Trace and Ghostbuster belong to this expanding effort to recover signatures of populations whose bones have not yielded DNA. Their promise is considerable, but so are their limitations. Several recent results remain preprints, and their conclusions depend heavily on assumptions about mutation rates, recombination, population size, and migration. A model can find a ghost because a ghost truly contributed DNA. It can also create one because the model is too simple. Imagine two ancestral populations remaining partly separated for hundreds of thousands of years, while exchanging genes occasionally. Their descendants may preserve deeply divergent lineages, even if no distinct archaic species ever entered the population. If a method assumes that the ancestors formed one freely mixing group, it may interpret that deep structure as a later episode of admixture. Simulations have shown that ignoring population structure can generate apparent interbreeding events that never occurred in the modelled history. This warning is central to the search for Ancestor X. A deep genetic branch is evidence of a complicated past, but it is not automatically proof of an unknown species. It may reflect introgression from an extinct archaic population, a long-isolated Homo sapiens group, structure among older ancestral populations, or several processes at once. No single explanation fully accounts for every signal currently reported. The debate becomes especially important in Africa. Ancient DNA preservation is poor across many tropical regions, leaving few archaic genomes for direct comparison. Modern African genomes preserve extraordinary diversity and very old lineages. Some analyses interpret portions of this variation as evidence that ancestors of present-day West African populations interbred with an unknown archaic group. Other models explain similar signals through a weakly structured ancestral stem, several African populations remaining connected over long periods, rather than one origin population receiving DNA from a separate species. Both possibilities challenge the older image of Homo sapiens emerging suddenly from one small birthplace. In one scenario, our ancestors met deeply diverged humans whose fossils remain unidentified. In the other, Homo sapiens formed through a network of populations spread across Africa, separating and reconnecting over immense spans of time. The difference matters, but both reveal that human origins were reticulate rather than linear. The most provocative models push that complexity deeper. One reconstruction suggests that two ancestral populations separated around 1.5 million years ago, and later contributed unequally to the population from which all living humans descend. Other models propose contact with deeply divergent, sometimes termed super-archaic, populations in both modern human and Neanderthal Denisovan history. These labels describe genetic distance, not officially recognized species. They cannot yet tell us whether the populations involved were Homo erectus, another named fossil form, or a lineage still hidden among fragmentary remains. A genome is therefore both archive and trap. It preserves encounters that bones may never reveal, but it can tempt researchers to turn statistical branches into fully imagined peoples before the evidence warrants it. Each signal must be tested against alternative models, independent methods, ancient genomes, proteins, fossils, and geography. Denisovans prove that a genetic ghost can eventually acquire teeth, jaws, and perhaps a face. Most figures emerging from our DNA still stand at the beginning of that journey. To understand where they belong, the genome must be placed back among the human lineages it connects. Denisovans were not isolated strangers appearing from nowhere. They were close relatives of Neanderthals, distant cousins of modern humans, and participants in a network of ancestry that repeatedly crossed the boundaries later drawn between species. Those boundaries become harder to defend the closer the genomes are examined. On a simplified diagram, modern humans, Neanderthals, and Denisovans appear as three branches descending from an older common population. The ancestors of modern humans separate first. Later, the lineage leading to Neanderthals and Denisovans divides again. This outline is broadly useful, but it hides almost everything that made their history real. Branches did not remain sealed. Populations changed size, moved across continents, split into regional groups, and met one another after long periods apart. The family tree repeatedly became a web. The common ancestors of Neanderthals and Denisovans probably separated from the lineage leading to modern humans hundreds of thousands of years before the populations recognized in the later fossil and genetic record. Estimates vary because mutation rates, generation times, and population models differ, but the division reaches deep into the Middle Pleistocene. Afterward, the Neanderthal and Denisovan ancestral lineages diverged from each other. Neanderthals became associated mainly with Western Eurasia, extending from Atlantic Europe into parts of Western and Central Asia. Denisovan-related populations appear to have occupied a much broader and still poorly mapped territory across Eastern Eurasia. Geography, however, was never permanent. Ice sheets advanced and retreated. Grassland corridors opened and closed. Forests shifted north and south. Deserts expanded, then weakened. Mountain systems created barriers but also valleys through which animals and humans could move. A population isolated for thousands of generations could later encounter relatives whose ancestors had departed long before. By then, their bodies, immune systems, and genetic variation had changed, but the separation was not always enough to prevent reproduction. The word species suggests a firm biological border. In human evolution, that border is often blurred. Neanderthals, Denisovans, and Homo sapiens were distinct populations with long independent histories, yet they were genetically compatible. Their descendants could survive and sometimes reproduce. This does not mean that every encounter was common, peaceful, or equally successful. The genome preserves outcomes, not social circumstances. It shows that contact occurred, but not whether meetings involved small migrating groups, absorbed outsiders, repeated partnerships, or rare events whose descendants later multiplied. Modern humans outside Africa generally carry some Neanderthal ancestry, inherited from encounters after populations of Homo sapiens expanded into Eurasia. Denisovan-related ancestry is distributed differently. It reaches its highest known proportions in parts of near Oceania, and is also present at lower levels in several Asian populations. The pattern cannot be explained by a single meeting in one location. Different modern populations carry different Denisovan segments, and some of those segments appear closer to the Altai Denisovan genome than others. This implies repeated contact with Denisovan populations that had already diverged from one another. The Altai genome is therefore not a perfect representative of every Denisovan who ever lived. It is one individual from one population, in one region and one period. Using it as the sole model for all Denisovan ancestry is like trying to reconstruct the genetic history of an entire continent from one person living near its edge. Recent analyses of near-Oceanian genomes strengthen the conclusion that at least 3 Denisovan-related sources contributed to the ancestry of populations in that region. Some branches may have been separated for hundreds of thousands of years before meeting modern humans independently. This diversity changes the meaning of the name. Denisovan is not necessarily a narrow biological category with one appearance and one way of life. It may describe a collection of related populations spread across environments ranging from the cold Altai to the high Tibetan Plateau, northern Chinese river systems, subtropical islands, and perhaps tropical Southeast Asia. Their bodies and cultures may have differed substantially. What united them genetically may have been no more uniform than the ancestry shared by widely separated modern human populations today. Neanderthals were also diverse. Fossils and genomes show regional and temporal variation, population replacements, and periods of severe isolation. Some Neanderthal groups were more closely related to Denisovans than others through later gene flow. Denisovan genomes contain evidence of Neanderthal ancestry, and certain Neanderthal genomes preserve contact with populations related to modern humans. These exchanges did not happen once at a single prehistoric crossroads. They occurred at different times and left unequal traces. One especially revealing pattern lies within the Denisovan genome itself. Parts of it appear to come from a deeply divergent hominin population, sometimes described in models as super-archaic. The term does not identify a species. It means that the inferred donor lineage had separated very early from the ancestors of the better sampled human groups. One proposal suggests that ancestors of Neanderthals and Denisovans met such a population in Eurasia more than 700,000 years ago. Homo erectus is often raised as an attractive candidate because early Homo populations had already spread widely across Eurasia, but no genetic evidence currently links the signal securely to that fossil name. The temptation to attach every genetic branch to a familiar skull is understandable. Paleoanthropology already contains names such as Homo erectus, Homo heidelbergensis, Homo antecessor, and other regional forms. Yet those names were built from anatomy, geography, and geological age, while genetic populations are reconstructed from patterns of inheritance. The two systems do not align automatically. A fossil population may contain ancestry from several genetic sources. A genetic lineage may include bodies that paleontologists would classify under more than one name. Scholars still debate where some Middle Pleistocene fossils belong, because evolution did not organize itself for the convenience of later taxonomy. The relationship among modern humans, Neanderthals, and Denisovans also challenges the language of replacement. Homo sapiens eventually became the only surviving human population, but this was not a total biological erasure. Neanderthals and Denisovans persist in fragmented form within later genomes. Their survival is uneven, filtered by natural selection and demographic history, but it is real. At the same time, saying they survive in us can become sentimental if taken too literally. A few inherited segments are not the same as the continuation of a people, their communities, or their cultural worlds. Genetic persistence and social survival are different things. Some archaic inheritance may have been useful. Denisovan-related variants have been associated with high-altitude physiology, immune regulation, metabolism, and other traits. Neanderthal-derived variants influenced immunity, skin biology, and aspects of physiology in populations outside Africa. Many other segments were removed, especially around regions important for reproduction and gene regulation. The pattern suggests both compatibility and cost. These populations were close enough to produce descendants, yet different enough that some combinations were selected against. This mixture makes the old ladder of progress impossible to defend. Neanderthals were not a failed rehearsal for modern humanity, and Denisovans were not an obscure side branch with no consequence. Nor did Homo sapiens move through an empty Eurasia, replacing static populations one after another. Expanding groups entered inhabited landscapes. They met people adapted to local climates, carrying knowledge developed across thousands of generations. Genes crossed between them, and cultural knowledge may have crossed as well. Though archaeology rarely allows the direction of influence to be proved, no single model fully accounts for every surviving pattern. Population structure can mimic admixture. Repeated interbreeding can resemble one prolonged event, and missing genomes leave large uncertainties. Still, the broad conclusion is secure. The major human lineages of late Pleistocene Eurasia were neither completely separate nor simply blended into one population. They remain distinct enough to preserve deep histories, yet connected enough to exchange ancestry. The clearest proof would not come from a population-level statistic, but from one person, in a fragment of bone from Denisova Cave. The two branches that diagrams kept apart would be found joined in the same body, with a Neanderthal mother and a Denisovan father. The abstract network would suddenly become a family. The family was discovered in a fragment so small that it might easily have disappeared into a tray of unremarkable bone. It came from Denisova Cave, but unlike the finger bone that first revealed an unknown population, this piece did not immediately announce who it belonged to. Its shape offered little help. The breakthrough came through a method designed to screen large numbers of fragmentary remains by their proteins, allowing researchers to identify human bone among the much more abundant remains of animals. Once the fragment was recognized as human, DNA transformed it into one of the most extraordinary individuals ever recovered from the Pleistocene. She is often known by the laboratory name Denisova-11 and more informally as Deni. Genetic analysis showed that her mother was Neanderthal and her father was Denisovan. She was not a distant descendant carrying a faint mixture from an encounter many generations earlier. She was a first-generation child of two deeply separated human populations. Half of her nuclear genome came from one lineage and half from the other, preserving in a single body the direct result of a meeting that diagrams had reduced to crossing arrows. Her maternal ancestry was unmistakably Neanderthal. Her mitochondrial DNA, inherited through the maternal line, fell within Neanderthal variation across her nuclear genome. One chromosome copy repeatedly matched Neanderthal sequences while the other aligned more closely with Denisovan ancestry. The pattern was too balanced and too recent to represent ordinary background admixture. Her parents themselves belonged to different populations. Even this simple statement hides complexity. The father was Denisovan, but his genome already contained some Neanderthal ancestry inherited from earlier contacts. The mother, meanwhile, was more closely related to later Neanderthals from Western Eurasia than to an older Neanderthal previously found in the same cave. This suggests that Neanderthal populations moved across large distances, and that the Altai region did not contain one stable, isolated community. Different Neanderthal groups entered or passed through the area at different times, meeting Denisovan populations whose own histories were equally mobile. Deni's existence does not prove that Neanderthal Denisovan families were common. One individual cannot reveal the frequency or social pattern of such unions. Yet her discovery among the tiny number of archaic individuals with usable genomes is difficult to dismiss as biologically negligible. If contact had been almost impossibly rare, finding a first-generation offspring in such a small sample would be extraordinarily fortunate. The evidence therefore suggests that when these populations encountered one another, interbreeding may not have been exceptional. The cave stood within a shifting contact zone. During some climatic periods, Neanderthal ranges extended eastward. Denisovan-related populations occupied the Altai and probably territories far beyond it. Changes in vegetation, prey distribution, and temperature could bring communities into the same valleys after generations of separation. A cave with reliable shelter and access to water would attract repeated occupation. The people who arrived did not know that later scientists would classify them into lineages. They encountered other humans who were different, but likely recognizable as people in the most immediate sense tool users, firekeepers, hunters, gatherers, and social beings navigating the same dangerous landscape. The genome cannot tell whether Deni's parents belonged to neighboring communities, whether one individual had joined another group, or whether the encounter was brief. It cannot recover their gestures, loyalties, or the way their child was regarded. Any attempt to describe those details would move beyond evidence. What survives in the record suggests only that genetic and social boundaries were permeable enough for a child to be born and to live long enough for her bones to enter the cave deposit. Her age at death has been estimated from skeletal development, but the fragmentary evidence limits precision. She was not an infant, and her remains do not preserve a life history in the way a complete skeleton might. There is no secure cause of death, no reconstructed face, and no burial that clearly marks how others treated her. The child safety limits of the surviving record are almost absolute here. Nothing in the evidence supports a violent narrative, and none is needed. The significance lies in her life, not in imagining her end. That life complicates the notion that hybrid individuals were biological dead ends. Deni herself left no known descendants, but other genomes show that Neanderthal-Denisovan admixture persisted through later generations. Denisovan populations carried Neanderthal-derived DNA, proving that at least some mixed individuals reproduce successfully. The same applies to encounters between archaic humans and Homo sapiens. Certain genomic regions were later selected against, especially where combinations may have reduced fertility or disrupted development, but enough ancestry survived to demonstrate repeated reproductive compatibility. The word hybrid can also mislead. It may evoke two sharply separate species meeting across an absolute divide. Neanderthals and Denisovans had been evolving apart for a long time, but they shared a relatively recent common ancestry and remained capable of producing viable descendants. Their difference was real, yet not complete. Deni is evidence of both facts simultaneously. Her chromosomes preserve deep separation on one level and immediate family connection on another. This duality runs throughout human evolution. Populations can be distinct without being isolated forever. They can develop characteristic anatomies and genetic histories while still exchanging genes at the edges of their ranges. A branching tree captures divergence, but only a network captures reunion. Deni embodies the place where those models meet. She also exposes the limits of fossil classification. Had her genome not survived, the small bone fragment could never have revealed that her parents belonged to two archaic populations. Even a complete skeleton might have been difficult to classify if its anatomy combined traits in unexpected ways. Paleoanthropologists often debate whether unusual fossils represent separate species, regional variation, mixed ancestry, or individual diversity. Deni demonstrates that mixed ancestry was not merely theoretical. Some ancient bodies truly combined lineages that taxonomic systems prefer to separate. This realization matters for fossils across Asia. A jaw may display a mosaic of robust and more derived traits. A skull may resemble one regional group in its face, and another in its cranial vault. Such combinations do not automatically prove interbreeding, but the possibility can no longer be treated as remote. Harbin, Siahi, Penghu, and other Middle Pleistocene remains must be considered within a world where populations diverged, moved, and sometimes rejoined. The social implications are even more elusive. Interbreeding requires proximity, but not necessarily cultural equality or long-term integration. A single migrant may be absorbed into another community. Small groups may merge during periods of demographic stress. Repeated contact may occur along ecological frontiers. Human communities are rarely sealed units, especially when populations are sparse and survival depends on alliances, knowledge, and access to territory. Yet no single explanation fully accounts for Deni's family. The genome records biological relationship without preserving the social world that made it possible. Her mother's Western affinities offer one further clue. Neanderthal populations were not fixed permanently in Europe or Western Asia. They could move eastward across the steppe and mountain corridors of Eurasia, entering regions occupied by Denisovans. The father's inherited Neanderthal ancestry shows that such meetings had happened before. Deni was therefore not the beginning of contact, but one visible moment within a longer pattern. That pattern helps explain why later human genomes appear layered. A Denisovan population could inherit DNA from Neanderthals, then contribute some of its own ancestry to Homo sapiens. Segments could pass through more than one lineage before reaching people alive today. An apparently Denisovan fragment might carry a deeper history involving earlier populations. Human ancestry becomes a sequence of nested encounters, each leaving only partial traces. Deni's bone turns that complexity into something immediate. She was not a statistical abstraction, or a ghost reconstructed from branch lengths. She was one person whose parents came from different human worlds. Her existence shows that the vanished populations of Eurasia did not merely replace one another across empty centuries. They met face to face. They recognized enough common humanity, or at least enough biological compatibility for their histories to join. From that cave, the question widens again. If such a family existed in the Altai, how many similar meetings occurred across the rest of Asia? The continent was enormous, its environments radically varied, and the Denisovan populations encountered by modern humans were not all closely related to the individual from Siberia. To find them, the search must leave the cave and follow the genetic shadows across mountains, river valleys, forests, islands, and tropical coastlines where almost none of their bones have survived. The genetic shadows lead away from the Altai in every direction, but the farther they travel, the less the landscape resembles the cold cave where Denisovans first became known. Asia during the Pleistocene was not one environment, and could not have supported one uniform population. It contained frozen mountain valleys, dry continental interiors, monsoon forests, broad river plains, high plateaus, subtropical coastlines, and islands repeatedly joined to or separated from the mainland as sea levels changed. If Denisovan-related people occupied even a fraction of this territory, their world was larger and more varied than the surviving evidence once allowed anyone to imagine. Denisova Cave lies near a natural crossroads between Siberia, Mongolia, Central Asia, and the mountain systems farther south. During colder periods, open steppe environments could connect distant regions, allowing large grazing animals and the humans who followed them to move across broad corridors. During warmer intervals, forests expanded and routes changed. The cave's inhabitants did not live at the edge of a permanently frozen wasteland. They occupied a dynamic zone where ecosystems repeatedly reorganized, creating opportunities for migration and periods of isolation. The Altai Denisovan genome represents only one point within this shifting geography. The ancestry carried by living populations shows that related groups must have existed much farther south and east. Some Denisovan-derived DNA in modern people is too different from the Altai sequence to have come directly from a population identical to the one sampled in Siberia. These segments suggest that Denisovan branches had separated from one another long before they encountered Homo sapiens. By the time modern humans moved through eastern and southeastern Asia, they may have met several populations whose last common ancestors with the Altai group lived hundreds of thousands of years earlier. This is the first major contradiction in the map. The best ancient nuclear genome comes from a northern cave, but the strongest surviving Denisovan ancestry occurs among populations in near Oceania, including New Guinea and neighbouring islands. The ancestors carrying that DNA had travelled through southern Asia and across stretches of sea. They could not have acquired all of it from one brief encounter near Siberia, unless later migrations followed an improbably indirect path. A more plausible picture places Denisovan-related groups across parts of eastern and southeastern Asia, occupying regions where ancient DNA preservation is especially poor. Warm climates are destructive archives. Moisture, acidity, microbial activity, and heat break down DNA rapidly. A cave in Siberia can preserve molecules that a tropical site loses within a much shorter span. The absence of Denisovan genomes from tropical Southeast Asia therefore does not demonstrate that Denisovans were absent. It may reveal only that the environment erased the evidence most useful to geneticists. Teeth and dense bones sometimes survive, but even these may yield proteins rather than DNA. Many fossils are fragmentary, and some were excavated long before Denisovans were known, then classified under broad or uncertain labels. The Tibetan Plateau provides one of the clearest indications that the Denisovan world extended beyond the Altai. At Baishia Karst Cave, high above sea level, a robust ancient jaw from Siahe was identified as Denisovan-related through protein analysis. Sedimentary evidence and archaeological remains indicate repeated human occupation of the plateau across long periods. Whoever lived there faced thin air, cold temperatures, intense seasonal variation, and limited resources. This was not a temporary extension into an easy landscape. It suggests populations capable of returning to or remaining within one of the most demanding environments inhabited by ancient humans. Farther east, the record has begun to acquire additional anchors. The Harbin cranium from northeastern China, more than 146,000 years old, was long difficult to place. Its combination of features encouraged competing classifications and even the proposal of a separate species. Molecular evidence later linked it closely to Denisovans through preserved proteins and mitochondrial DNA recovered from dental calculus. The result does not mean every unusual Middle Pleistocene fossil in China is Denisovan, but it shows that a population once known almost entirely from a Siberian genome had a physical presence far beyond the Altai. To the south, the Penghu 1 mandible from the waters near Taiwan extends the possible range into a subtropical setting. Protein evidence from tooth enamel identifies the jaw as belonging to a Denisovan male. The landscape he inhabited would have looked very different depending on sea level. During glacial periods, lowered seas exposed broad coastal plains and altered the distance between Taiwan and the Asian mainland. River systems crossed areas now submerged. Grasslands, wetlands, and coastal ecosystems created migration routes that no longer exist on modern maps. Penghu is important because it breaks the image of Denisovans as a cold-adapted mountain population, a human group associated with Siberian caves and the Tibetan Plateau also reached, or was represented within, the warmer margins of East Asia. Denisovan-related populations may have tolerated an extraordinary environmental range. That adaptability could have emerged through cultural flexibility, regional biological adaptation, or the fact that Denisovan included several populations with different histories rather than one ecologically uniform people. Southeast Asia adds another layer of possibility. Fossils from the region reveal a crowded human landscape. Homo erectus survived on Java for an immense span of time. Homo floresiensis lived on Flores. Homo luzonensis occupied the Philippines. Modern humans eventually moved through the region, while Denisovan ancestry in present-day populations shows that they encountered Denisovan-related people somewhere along their routes. The exact locations remain uncertain. No fossil from these islands has yet provided the secure genomic connection needed to identify the population that contributed the strongest Denisovan ancestry to Papuan and other Oceanian ancestors. This missing encounter may have occurred on the Asian mainland, on the now submerged Sunda Shelf, in Wallacea, or across more than one region. During periods of low sea level, much of western Indonesia formed a broad extension of continental Southeast Asia. Rivers crossed land now beneath the South China Sea and Java Sea. Farther east, however, deep water channels remained. Reaching Sahul, the combined Ice Age landmass of Australia and New Guinea, required intentional sea crossings. The modern human groups who made these journeys carried Denisovan-derived DNA, but whether they acquired it shortly before crossing the islands or earlier in continental Asia remains debated. The genetic evidence points to multiple contacts. Some East Asian populations carry Denisovan-related ancestry resembling one source, while near-Oceanians preserve contributions from several deeply separated sources. Ancient genomes add further complexity. Certain early East Asians carried more Denisovan ancestry than many later populations, while other lineages, including ancestors related to the Jomon, appear to have had little direct contact. Human movement through Asia was not one wave passing through one corridor. Different groups followed different routes, encountered different populations, and later mixed with one another, obscuring the original geography. The continent also contained regions from which almost no ancient human DNA survives. Southern China, mainland Southeast Asia, the drowned plains of Sunda, and many tropical islands remain large blank areas. Those blanks should not be filled carelessly. It would be easy to assign every unexplained fossil or genetic segment to Denisovans, but the evidence does not support such certainty. Other human populations lived in Asia, and some may have contributed ancestry without belonging to the Denisovan line as currently understood. Scholars still debate how many archaic groups were present, and how their anatomical names correspond to genetic populations. What survives in the record suggests a network of regional Denisovan-related communities, rather than a single population spreading unchanged from Siberia to the tropics. Northern groups may have adapted to strong seasonality and cold. Plateau populations confronted low oxygen. Eastern coastal communities occupied river plains and subtropical environments. Southern populations, if the genetic evidence is read correctly, may have lived in monsoon forests and island landscapes. Their technologies, diets, bodies, and social networks likely varied as much as their habitats. Such variation helps explain why their fossil identity remained hidden. Researchers were searching for a recognizable Denisovan type before enough anatomy existed to define one. Yet a population complex extending across Asia might not produce one consistent appearance. Regional groups could retain shared ancestry while developing different traits, especially after long isolation and occasional mixture with other humans. A jaw from Tibet, a skull from northeastern China, and a mandible near Taiwan need not look identical to belong within related Denisovan histories. The map is therefore being reconstructed backward. Genomes in living people indicate that encounters happened. Proteins and fragmentary DNA begin to attach those encounters to real fossils. Submerged landscapes and ecological corridors suggest how populations could have moved. But enormous distances remain between the known points, and the people who once occupied them are still largely absent from the ground. For years, Denisovans seemed almost bodiless, a genome in Siberia with descendants thousands of kilometers away. That condition is finally changing. Teeth, jaws, a rib, a massive skull, and molecular traces preserved where ordinary DNA failed are beginning to give the genetic ghost an anatomy. The face is still incomplete, but it is no longer entirely invisible. The ghost is beginning to acquire a body, but not in the clean, orderly way early reconstructions once imagined. There is no single Denisovan skeleton laid out beneath museum lights, no complete sequence of skull, spine, pelvis, hands, and feet from which anatomy can be confidently rebuilt. Instead, the emerging portrait is assembled from distant pieces: enormous molars in Siberia, a robust jaw from the Tibetan Plateau, a mandible from the waters near Taiwan, a rib from a high-altitude cave, and a nearly complete skull from northeastern China, whose molecular identity was hidden for more than a century of accumulated dust and uncertainty. Each piece adds substance, none yet closes the debate. The earliest Denisovan fossils offered almost no face at all. A finger bone could reveal ancestry but little appearance. The molars from Denisova Cave were more suggestive. They were large, with thick enamel and complex roots, strikingly different from the smaller teeth typical of recent Homo sapiens. Their size hinted at a robust chewing apparatus, but teeth are treacherous witnesses. Large molars can reflect retained ancestral traits. regional adaptation, developmental variation, or dietary demands. They do not automatically define the shape of the skull around them. For years, the question remained almost absurdly unbalanced. Geneticists could compare a high-quality Denisovan genome with populations across the world, while anatomists could not say with confidence what a Denisovan face looked like. Some researchers attempted cautious reconstructions from patterns of gene regulation, estimating skeletal traits through methylation differences preserved in ancient DNA. These efforts suggested a broad face, a robust jaw, and other features unlike those of both Neanderthals and modern humans. Yet such reconstructions remain indirect. They are predictions derived from molecular patterns, not substitutes for bones. The Xiahe mandible changed the scale of the evidence. Found in Baishia Karst Cave on the Tibetan Plateau, it preserves much of a lower jaw and two large molars. The bone is thick and powerful, lacking a modern human chin. Its shape belongs comfortably within the robust world of Middle Pleistocene Homo, but anatomy alone could not identify it as Denisovan. No recoverable nuclear DNA was required. Ancient proteins preserved in the dental tissue carried enough evolutionary information to connect the individual with the Denisovan lineage. This use of paleoproteomics opened a new path. Proteins survive longer than DNA under many conditions. Their sequences contain fewer informative differences, so they cannot usually reconstruct population history with the same resolution as a genome, but they can identify broad evolutionary relationships where DNA has vanished. For Asian fossils from warm or ancient deposits, this may be the only molecular bridge between anatomy and ancestry. The Xiahe jaw revealed that Denisovan-related people occupied the Tibetan Plateau long before modern populations established their later genetic history there. It also offered one of the first substantial anatomical clues, a deep, robust mandible capable of supporting large teeth. Yet even here, caution is necessary. One jaw does not represent all Denisovans across Asia. It may reflect a local high-altitude population, an earlier stage of Denisovan evolution, or a mixture of inherited and regional traits. Additional remains from Baishia Karst Cave deepen the connection. A fragmentary rib identified through molecular analysis belonged to a Denisovan individual and came from deposits associated with long-term occupation of the plateau. A rib cannot reveal a face, but it places a Denisovan body within a particular environment and archaeological sequence. It also demonstrates that the Xiahe mandible was not an isolated traveller accidentally preserved far from the main population. Denisovan-related people returned to or persisted in the region across substantial spans of time. The Penghu 1 mandible adds a different kind of body. Dredged from the seafloor near Taiwan, it comes from a landscape now submerged. Its precise age remains difficult to establish, because it was recovered outside a secure archaeological layer, but the jaw itself is unmistakably robust. Tooth enamel proteins identify the individual as a Denisovan male. This molecular sex determination is a lesser-known but remarkable detail. Proteins in enamel can preserve signatures associated with sex chromosomes, even when DNA no longer survives. Penghu's jaw shares some broad features with the Xiahe mandible, including great thickness and powerful construction. Yet it is not simply a duplicate. The two individuals lived in radically different environments and may have belonged to populations separated by geography and time. Their similarities could reflect shared Denisovan ancestry, while their differences may record regional evolution. The evidence remains incomplete, but it now becomes possible to speak of Denisovan anatomy as more than a set of theoretical predictions. The most dramatic candidate for a Denisovan face is the Harbin cranium. The skull is large and unusually complete, with a low cranial vault, massive brow region, broad face, and large teeth. Its history of discovery is tangled, and its original geological context was not documented with modern archaeological precision. For years, researchers debated whether it represented a new species, a regional form of Middle Pleistocene Homo, or a close relative of other enigmatic East Asian fossils. Then molecular evidence began to resolve what shape alone could not. Proteins recovered from the cranium showed its closest known relationship to Denisovans. More surprisingly, mitochondrial DNA was recovered not from the dense temporal bone or from a tooth, where such efforts had failed, but from dental calculus adhering to the teeth. Mineralized plaque had trapped biological material from the individual's mouth and preserved enough mitochondrial DNA to place the sequence within Denisovan variation. The method itself may prove nearly as important as the identification. Dental calculus has long been used to study diet, oral microbes, and environmental particles. Harbin demonstrated that it can sometimes preserve the individual's own DNA when the skeleton does not. Fossils previously considered genetically silent may therefore contain molecular evidence in places researchers once treated as secondary deposits. Harbin offers something the cave fragments could not: a nearly complete cranium that may belong to a Denisovan-related individual. It gives the ghost a brow, a cranial vault, cheekbones, and a broad facial structure. Yet one skull cannot define a continent-wide population complex. The Harbin individual may represent a northern Denisovan branch, while southern and plateau populations developed different appearances. Denisovan-related ancestry already appears deeply structured in modern genomes, so anatomical diversity should be expected. This complicates species classification. Some researchers prefer to group Denisovan fossils within an existing species name. Others regard Denisovans as a distinct evolutionary population that may deserve its own formal taxonomic designation. The problem is that genetic categories and fossil species do not map neatly onto one another. Denisovans could include fossils previously assigned to several anatomical groups. Conversely, not every robust East Asian fossil belongs to them. Names such as Homo longi have been proposed for Harbin, while other fossils are placed within Homo daliensis, archaic Homo sapiens, late Homo erectus, or broader Middle Pleistocene groupings. A molecular connection to Denisovans does not automatically settle which anatomical name should take priority. Taxonomy depends on definitions, type specimens, comparisons, and disagreements over how much variation belongs within one species. There's also the possibility of mixture. If Denisovan populations interbred with Neanderthals, modern humans, and perhaps more deeply divergent groups, their anatomy may have been mosaic as well as regionally variable. A skull combining ancestral and derived features need not represent a clean intermediate stage. It may preserve a history of repeated population contact. What survives now suggests a broad pattern: powerful jaws, large teeth, substantial facial and cranial robusticity, and enough regional variation to resist a single simple reconstruction. But almost everything below the head remains obscure. Their stature, limb proportions, hand anatomy, locomotion, and sexual dimorphism are still poorly known. The famous Denisovan portrait remains far less complete than that of Neanderthals. Even so, the transformation is unmistakable. Denisovans are no longer only a genome from a finger bone. They now occupy caves in Siberia and Tibet, appear in a massive skull from northeastern China, and stand on vanished coastal plains near Taiwan. Molecular evidence is gradually joining bones that once seemed unrelated. The ghost is not fully embodied, but it has begun to look back at us. And once a face becomes possible, the next question is unavoidable. What did these people actually do across such different worlds? The answer must come not from genes alone, but from hearths, butchered bones, stone tools, worked materials, and the fragile traces of daily life left in the caves they repeatedly entered. The caves they entered were not homes in the modern sense. They were shelters used, abandoned, revisited, and altered by many generations. Fires burned in some chambers, animals dragged bones into others, sediments shifted under feet, water, frost, and burrowing creatures. A stone flake could remain near the place where it fell, or move slowly through a deposit until its original association became uncertain. Reconstructing Denisovan daily life therefore requires patience. The evidence does not preserve one intact campsite frozen at a single moment. It preserves repeated occupations compressed into layers, often shared with Neanderthals and perhaps later modern humans. At Denisova Cave, the archaeological sequence contains thousands of stone artifacts and animal remains. Some tools were made from local raw materials gathered near the valley, others used stone transported from farther away, suggesting movement across a wider territory or exchange between groups. Flakes, scrapers, pointed pieces, and prepared cores show that the cave's occupants understood how to control fracture, maintain sharp edges, and plan several steps ahead. Yet assigning particular technologies to Denisovans is difficult, because different human populations use the cave at different times, and because some layers have been disturbed. This uncertainty matters most when the artifacts appear unusually elaborate. The cave has produced ornaments, including beads and a polished stone bracelet, that once encouraged dramatic claims about Denisovan symbolic culture. The objects demonstrate sophisticated workmanship somewhere within the cave's long history, but their makers cannot always be identified securely. Some may belong to early modern humans, others may be associated with Denisovan occupation. Scholars still debate how objects moved through the deposits, and whether the dating of the artifacts and human remains truly overlaps. The safest conclusion is not that Denisovans lacked symbolic behaviour, nor that every ornament was certainly theirs, but that the evidence remains incomplete. The simpler objects reveal more than they first appear to. A sharp flake is an extension of the hand. It can remove hide, cut flesh, shape wood, slice plants, and prepare other materials that rarely survive. A scraper may indicate hide working, but it can also perform several tasks depending on its edge and use history. The stone tools remaining in a cave are only the durable portion of a larger technological system. Wooden shafts, carrying containers, fibres, bindings, clothing, and bedding may have formed much of Denisovan material culture while leaving almost no trace. Animal bones provide another archive. At Denisova Cave, the remains include large and small mammals adapted to changing environments around the Altai. Some bones carry cut marks left when stone edges removed flesh. Others were broken while fresh, possibly to extract marrow or grease. Burned fragments show contact with fire, though not every burned bone came from deliberate cooking. Carnivores also use the cave, and their tooth marks can overlap with human modification. Archaeologists distinguish these traces by their shape, location, microscopic structure, and relation to the surrounding deposit. What survives suggests that the cave's human occupants exploited a wide range of animals rather than depending on one species. They lived in landscapes inhabited by horses, bovids, deer, wild sheep or goats, and other game that shifted with climate. Hunting such animals required knowledge of terrain, seasonal movement, wind, and animal behavior. It probably involved cooperation, although the size and organization of hunting groups cannot be recovered directly. Some carcasses may have been killed by humans, others may have been scavenged from predators or found after natural deaths. No single explanation fully accounts for every assemblage. Large prey offered meat, fat, hides, sinew, bone, and horn. Each material demanded different knowledge. Hides could become insulation, coverings, sleeping surfaces or containers. Sinew could bind tools. Bone could be shaped into points, retouchers, or implements whose purpose is not always obvious. At Denisova Cave, fragments of animal bone show deliberate working beyond simple butchery. Some were used to retouch stone edges, pressing or striking flakes from a tool to sharpen and reshape it. Such maintenance reveals a practical economy in which objects were repeatedly repaired rather than discarded after one use. At Bashir Karst Cave on the Tibetan Plateau, the evidence reflects a different environmental challenge. The inhabitants processed animals adapted to high elevations, including large herbivores that could provide calories, hides, and fuel in a cold, treeless landscape. Cut marks, percussion damage, and bone tools indicate that Denisovan-related people did more than pass briefly through the cave. They knew how to use plateau resources and returned across long periods. Some animal remains may represent hunting, while others could have been collected from natural deaths or carnivore kills. The pattern is one of flexibility rather than dependence on a single strategy. Fuel would have been a constant concern. In forested valleys, wood was available, though gathering it required labour. On high plateaus, shrubs, dung, bone. And whatever combustible material the environment offered may have supplemented scarce wood. Fire provided warmth, light, protection from predators, and a way to transform food. It dried wet clothing and hides, hardened wooden points, and created a social centre within dark shelters. Yet hearths are often difficult to associate with a particular population unless they are securely dated and linked with identifiable remains. The Denisovans' food was almost certainly broader than the surviving bones imply. Plants rarely preserve in the same dramatic quantities as teeth or large mammal remains. Roots, seeds, nuts, berries, leaves, fungi, and edible inner bark may have contributed substantially depending on region and season. In subtropical or tropical areas, plant foods could have been especially important. The absence of preserved vegetation does not justify imagining a diet made entirely of meat. Dental calculus, microscopic residues, and wear patterns may eventually recover more of this missing dietary world. Regional diversity again becomes central. A Denisovan group in the Altai would have faced strong winters and seasonal prey movement. A plateau population confronted thin air, cold, and limited plant growth. A community on exposed coastal plains near Taiwan could exploit rivers, wetlands, shorelines, and subtropical vegetation. Southern Denisovan-related populations may have lived among monsoon forests where visibility was short. Plant resources abundant, and hunting conditions unlike those of open steppe. It would be misleading to speak of one Denisovan way of life across all of Asia. Their equipment must have changed with these environments. Heavy cutting tools useful for processing large carcasses were not the only solution. In forests, smaller portable tools and wooden technologies may have mattered more. On the plateau, durable clothing, shelter construction, and fuel management were essential, near coasts, fishing, shellfish collection, watercraft, or shoreline gathering become possibilities. But direct Denisovan evidence for these behaviors remains absent. The genetic maps suggest where encounters occurred, not which tools were carried there. Population size also shaped daily existence. Denisovan genomes indicate periods of relatively small effective population size and separation between regional groups. In practical terms, many communities may have been small and scattered across vast territories. Meeting unrelated people could be rare but important. Information, mates, raw materials, and techniques may have moved through networks extending beyond a single camp. Long isolation could also cause regional traditions to diverge, helping explain why no single archaeological industry can easily be labeled Denisovan across Asia. The boundaries between cultural traditions and biological populations were probably porous. Neanderthals and Denisovans met in the Altai. Modern humans later encountered Denisovan-related groups elsewhere. Tools may have spread through observation, exchange, migration, or independent invention. Similar stone technologies do not prove that the same population made them, and different technologies do not always indicate different species. Archaeology records behavior, while genetics records ancestry. The two histories overlap, but they are not identical. This distinction prevents Denisovans from becoming either primitive shadows or imaginary masters of every unexplained artifact. They possessed the knowledge required to survive across demanding landscapes. They processed animals, maintained tools, controlled fire, and adapted their material lives to local resources. Their community endured long enough to diversify across a continent. Beyond that, caution remains necessary. Their rituals, languages, art, and social rules are mostly beyond recovery. What can be reconstructed is a quieter competence. Someone selected a suitable stone and struck it at the angle required to release a sharp flake. Someone carried fuel into a dark cave. Someone learned when animals crossed a valley or where edible plants returned after winter. Someone repaired a worn edge rather than beginning again. These acts rarely create spectacular museum objects, but they are the foundation of survival. On the Tibetan Plateau, that competence was tested more severely than almost anywhere else. The Denisovan-related people who lived there entered an environment where every breath carried less oxygen, where cold and elevation pressed against the body, and where adaptation had to occur through both culture and biology. Their presence in the thin air would eventually leave one of the clearest Denisovan traces in living humans. The clearest surviving trace of Denisovan life on the Tibetan Plateau does not lie only in the cave floor. It also survives in the physiology of people who live there now. At high altitude, the atmosphere contains the same proportion of oxygen as at sea level, but lower air pressure means that each breath delivers fewer oxygen molecules to the body. For newcomers, the consequences can be immediate headache, fatigue, disturbed sleep, shortness of breath, and in severe cases dangerous illness. Long-term survival requires more than endurance. It demands cultural knowledge, developmental adjustment, and, across many generations, biological adaptation. The Tibetan Plateau rises across a vast region often called the roof of the world. Much of it stands above 4,000 meters. Temperatures can fall sharply after sunset. Winds strip heat from exposed skin, and growing seasons are short. Water may be locked in ice, fuel can be scarce, and the landscape offers little shelter unless caves, rock overhangs, or constructed windbreaks are available. To imagine Denisovan-related people living here is to abandon the idea that archaic humans survived only in mild refuges. They entered a place where every ordinary task demanded more energy. Baishia Cave occupies one of the plateau's most important archaeological settings. The Shihi mandible established a Denisovan connection through ancient proteins, while later molecular work identified additional Denisovan remains and environmental DNA from the cave's deposits. Stone tools, butchered animal bones, and long sequences of occupation indicate that people returned to this location over immense spans of time. The cave was not simply visited once during an accidental excursion. It formed part of a repeatedly inhabited high-altitude landscape. This repeated presence matters because altitude is not a challenge solved by one invention. Clothing helps against cold, fire provides warmth, and hunting knowledge secures food, but none of these increases the amount of oxygen entering the blood. The body responds to low oxygen by changing breathing, circulation, and red blood cell production. Some responses help briefly, but create problems if sustained. Excessively high hemoglobin can thicken the blood and strain the cardiovascular system. Populations with deep histories at altitude often show physiological patterns that reduce these costs. Among present-day Tibetans, one of the best-known genetic associations involves EPAS1, a gene active in the body's response to low oxygen. A particular set of variants is strongly linked to Denisovan-related ancestry. The inherited sequence does not come from the Altai individual in a perfectly identical form, and the precise donor population remains uncertain, but its archaic origin is well supported. Natural selection later increased its frequency in Tibetan populations because it helped regulate the response to hypoxia in a way suited to life at elevation. This is one of the clearest examples of adaptive introgression in human evolution. A population does not need to invent every useful biological solution independently. When two lineages meet and have descendants, a variant shaped in one population can enter another. If the new environment makes that variant beneficial, selection can preserve and amplify it. The pattern may remain visible tens of thousands of years after the donor population has disappeared. Yet the story should not be reduced to a single high-altitude gene. EPAS1 functions within a wider system, and altitude adaptation involves many genes, physiological traits, and developmental processes. Cultural practices are equally important. Knowledge of seasonal routes, shelter, fuel, clothing, food storage, and animal behavior may have allowed people to enter the plateau before strong genetic adaptation became common. Genes can improve survival, but they do not replace experience. It is also uncertain where the Denisovan-related variant was first selected. The simplest narrative places its evolution directly on the Tibetan Plateau, where Denisovans lived before modern humans. That is plausible, but not fully demonstrated. The variant may have arisen in a related high-altitude population elsewhere, or existed at low frequency before becoming useful on the plateau. The timing of introgression into Homo sapiens remains debated, as does the location where the relevant populations met. The Jiahe mandible and later remains nevertheless make one possibility especially compelling. Denisovan-related people occupied high-altitude environments long enough to develop regional adaptations, and modern humans later inherited part of that biological history. This does not mean the plateau was continuously occupied by one unchanged population. Climatic shifts could empty regions for long intervals. Groups may have moved between lower valleys and higher terrain, replaced one another, or mixed. The archaeological layers compress these movements into a long but uneven record. The animals processed at Baishia reveal how survival may have worked. Large herbivores provided meat, marrow, hides, sinew, and bone. Their seasonal movements would have structured human movement. Hunting on open ground required reading distance, wind, and terrain, while scavenging could also supply valuable resources. Some bones were modified into tools, showing that carcasses were used beyond immediate consumption. A plateau population that wasted little had a better chance of enduring winter. Fire was another form of adaptation, but fuel may have been unreliable. People could gather shrubs, wood from lower valleys, dung, or other combustible material. A maintained fire meant more than warmth. It allowed frozen food to be processed, hides to be dried, tools to be repaired, and social life to continue after dark. Inside a cave, smoke left a residue of repeated occupation, though those traces are not always easy to assign to one population. Clothing must have been essential, yet almost none survives. Denisovans could not remain on the plateau through severe cold without insulation. Hides may have been wrapped, tied, scraped, softened, and perhaps stitched, although direct evidence for sewing is limited. The absence of garments is a preservation problem, not proof of simplicity. Much of the technology most necessary for high-altitude survival was organic and therefore nearly invisible archaeologically. The body itself would also have been shaped by elevation. Individuals born and raised on the plateau developed differently from newcomers, even without inherited adaptation. Breathing patterns, circulation, chest development, and metabolism respond to the environment. Denisovan populations remaining there over many generations may have combined developmental flexibility with genetic change. The robust Siahi jaw tells little about lungs or blood, but the inherited APAST signal offers a rare glimpse into physiology. This creates an unusual bridge between fossil, genome, and living population. A mandible recovered from a cave identifies Denisovan presence. Molecular evidence in modern humans identifies a useful archaic contribution. The high plateau provides the ecological setting linking them. Few episodes in deep human history can be traced across such different archives. Even here, however, the bridge is incomplete. The Denisovan population that contributed the adaptive sequence may not be identical to the individuals found at Baishia. Modern Tibetans received most of their ancestry from Homo sapiens populations with complex histories of their own. Later migrations, mixture, and selection reshaped the genetic landscape. The Denisovan contribution is important precisely because it is limited a small inherited segment with a large effect, preserved within an overwhelmingly modern human genome. The broader lesson is that disappearance does not erase every adaptation. A population can vanish as a recognizable community while one useful variant continues through another lineage. The people who first endured the thin air are gone, but part of their biological response may still help others breathe there. That legacy also changes how Denisovan expansion must be imagined. These were not humans confined to one northern cave. Their relatives occupied elevations that challenge even well-equipped travelers today. If they could adapt to the plateau, they may also have entered warmer forests, river plains, and coastal worlds farther south. The next traces appear not in one fossil bed, but along the routes taken by modern humans through Asia, where different groups encountered different Denisovan populations and carried fragments of them toward the islands beyond the continent. Beyond the high plateau, the Denisovan trail descends into warmer worlds where the evidence becomes both richer and more elusive. Rivers widen, forests thicken, coastlines shift with the ice ages, and the cold conditions that preserved DNA in Siberia give way to heat and moisture that rapidly destroy it. Here, Across eastern and southeastern Asia, Denisovan-related populations may have lived for hundreds of thousands of years while leaving almost no recoverable genome behind. Their presence must be reconstructed from scattered bones, ancient proteins, drowned landscapes, and the fragments of ancestry carried by people whose forebears later moved through the region. The Harbin cranium anchors one part of this southern and eastern expansion. Found in northeastern China, it belonged to a world of broad river valleys, seasonal cold, and ecological corridors connecting the interior of Asia with the Pacific margins. Molecular evidence places the individual close to Denisovans, transforming what had been an anatomically puzzling skull into a possible representative of a northern East Asian Denisovan population. Yet Harbin also warns against imagining that every Denisovan looked like the person whose skull survived there. Farther south, different climates, diets, and long periods of isolation may have produced bodies that varied considerably. The Penghu mandible carries the trail towards subtropical Asia. Today, the fossil's discovery location lies beneath water in the Taiwan Strait, but during glacial periods the sea retreated and exposed a vast lowland between the Asian mainland and Taiwan. Rivers crossed this plain. Herd animals moved through open habitats, freshwater wetlands, coastal resources, and patches of woodland created a landscape far more extensive than the narrow modern shoreline suggests. A Denisovan male lived somewhere within this vanished country, or near its margins, leaving behind a powerful jaw that survived after the land itself disappeared beneath rising seas. The submerged setting matters because many routes used by ancient humans no longer exist on present-day maps. During the Pleistocene, global sea level repeatedly fell by more than 100 meters. The Sunda Shelf joined much of western Indonesia to mainland Southeast Asia, creating a broad continental region containing river systems, forests, grasslands, and lakes. Modern seas cover these migration corridors, campsites, and possible fossil deposits. If Denisovan-related populations occupied them, some of the strongest evidence may now lie under layers of marine sediment. Farther east, deep channels continue to separate islands even when sea level was lowest. Wallacea, the island zone between continental Sunda and the ancient landmass of Sahul, was never fully joined by dry land. Crossing it required movement over water. Modern humans eventually travelled through this region and reached Sahul, carrying Denisovan ancestry with them. The great unresolved question is where that ancestry entered their populations. It may have been acquired on the Asian mainland, somewhere on the drowned Sunda Shelf within Wallacea, or through several encounters spread across different regions. The modern genetic map favours complexity rather than one meeting. Populations in East Asia carry Denisovan-related segments that are not identical to those found at high levels in Papua New Guinea and neighbouring parts of Oceania. Recent analyses indicate that ancestors of near-Oceanian communities received DNA from at least 3 populations related to Denisovans. These donor groups had already diverged deeply from one another. Their separation suggests long histories in different regions before Homo sapiens encountered them. One Denisovan source may have been more closely related to the genome recovered from the Altai. Others were far more distant, implying populations that had been isolated in southern or eastern Asia for immense spans of time. They need not have belonged to separate, formally recognized species. They may instead have formed a broad Denisovan population complex divided by mountain systems, deserts, forests, coastlines, and islands. Occasional contact could preserve a shared ancestry while still allowing strong regional differentiation. This possibility reshapes the meaning of tropical fossils. Southeast Asia already contains evidence for several ancient human populations. Homo erectus survived on Java until remarkably late in the Pleistocene. Homo floresiensis evolved on Flores, displaying a distinctive combination of small body size and ancient anatomical features. Homo luzonensis lived in the Philippines and is known from a limited set of bones and teeth. These populations demonstrate that island and tropical Asia supported multiple human lineages, some isolated for long periods. None can simply be declared Denisovan. The fossils of Homo floresiensis and Homo luzonensis have not yielded genetic evidence linking them directly to the Denisovan ancestry found in modern populations. Their anatomy also raises evolutionary histories that may reach deeper than the Denisovan lineage. Homo erectus remains an equally tempting but unconfirmed participant in the region's genetic story. The evidence remains incomplete, and assigning every unknown Asian fossil to Denisovans would replace one mystery with an unsupported certainty. Still, the crowded landscape makes encounters plausible. Denisovan-related groups moving south may have met populations whose ancestors had occupied Asia since much earlier dispersals from Africa. They may have exchanged genes with some of them, just as they did with Neanderthals and Homo sapiens. Deep genetic signals found within Denisovan ancestry have encouraged models involving contact with even older Eurasian populations. These inferred donors are sometimes called superarchaic, but the term describes their genetic distance rather than their anatomy or species identity. A southern Denisovan population could therefore have carried a layered inheritance before it ever met modern humans. Its genome might include ancestry from northern Denisovans, older Asian Homo populations, and regional groups that remain completely unknown. When fragments from such a population entered Homo sapiens, they would carry that deeper history forward. What appears today as one Denisovan segment may contain traces of several earlier population encounters nested within it. The environment would have shaped these southern groups differently from their relatives in Siberia or Tibet. Monsoon forests demand a different kind of movement from open steppe. Visibility is limited. Large animals are harder to track across dense vegetation. Plant foods may be abundant but highly seasonal. Rivers and coasts provide fish, shellfish, reptiles, birds, and wetland resources. Tools made from bamboo and wood could be more practical than heavy stone implements. Yet these materials rarely survive. This preservation bias creates the illusion of technological absence. A population relying heavily on organic tools may leave fewer recognizable artifacts than one working in stone. Bamboo can be split into sharp edges. Wood can become digging sticks, shafts, clubs, containers, and frames. Fibers can be twisted into cordage, nets, or bindings. None of these technologies requires a simple mind, and nearly all can vanish without trace in tropical soils. Clothing and shelter would also change. Thick hides essential in the Altai might become burdensome in humid forests. Lightweight coverings, rain protection, raised sleeping surfaces, and temporary shelters could matter more. Fire would remain useful, but maintaining it through heavy rain posed different challenges from preserving heat through a Siberian winter. Knowledge of poisonous plants, seasonal fruiting, river behavior, and tropical disease would become central to survival. Biological adaptation may have followed. Denisovan-derived variants in modern populations have been associated with immune functions and responses to pathogens. A major study of near-Oceanian genomes identified thousands of archaic variants with the potential to influence gene activity, including pathways connected to immune signaling. Some may have been retained because they helped descendants confront local diseases. Others may have persisted by chance or carried effects that remain uncertain. These inherited variants should not be treated as proof that Denisovans were uniquely resistant to tropical illness. Functional predictions are not the same as reconstructed prehistoric health. A variant may influence gene expression without producing a simple benefit, and its effect can depend on the surrounding genome and environment. Still, the concentration of archaic ancestry in immune-related regions suggests that contact with locally adapted populations sometimes gave migrating Homo sapiens useful biological material. The southward story is therefore not simply a migration across a map. It is a passage through different ecological worlds, each capable of separating populations and shaping them in new directions. A Denisovan community near the Taiwan Strait may have shared deep ancestry with people on the Tibetan Plateau while living a life almost unrecognizable to them. Farther south, related groups may have occupied forests and drowned plains whose archaeological record has barely been sampled. Modern genomes preserve the clearest evidence that these populations existed. The strongest Denisovan ancestry in Oceania cannot be explained by the few fossils currently known. It points toward encounters with people whose bodies remain undiscovered or unrecognized. Some early modern humans moving across Asia met Denisovan-related groups repeatedly. Others traveled along routes where such contact was limited or absent. The continent functioned not as one corridor but as a maze of branching paths. By the time Homo sapiens approached the islands leading towards Sahul, they were not entering an empty frontier. They carried ancestry from earlier encounters, and the human populations ahead of them possessed histories reaching far deeper into Asia. Somewhere in these warmer worlds, lineages met again. The genetic consequences would survive long after the places, communities, and circumstances of those meetings had vanished. The circumstances of those meetings have disappeared, but their timing can still be approached through the broken inheritance they left behind. Homo sapiens did not enter Asia in one movement made by one uniform population. Groups expanded along different routes, paused in some regions, disappeared from others, and mixed with communities they encountered along the way. By the time some of their descendants reached the islands between Zunda and Sahul, they already carried genetic evidence of encounters with Denisovan-related people. Yet those encounters were not all with the same Denisovan population, and they probably did not occur in one place or one brief episode. The earliest Homo sapiens populations moving beyond Africa had already entered a human world shaped by earlier migrations. Neanderthals occupied much of western Eurasia. Denisovan-related populations were distributed across parts of Asia. Other groups, perhaps descendants of much older expansions, survived in regions where their fossils remain difficult to classify. Modern humans were newcomers only in a regional sense. The continent had been inhabited by other forms of humanity for hundreds of thousands of years. Genetic evidence indicates that the ancestors of most people living outside Africa encountered Neanderthals relatively early in their expansion through Eurasia. Denisovan admixture followed a more geographically uneven pattern. Some populations acquired little, others inherited substantial amounts. This unevenness implies that Homo sapiens groups separated from one another before or during their encounters with different Denisovan communities. A meeting experienced by one migrating population might leave no trace in another population travelling only a few hundred kilometres away. The simplest old model imagined one Denisovan admixture event somewhere in Southeast Asia, after which the mixed population continued toward New Guinea and Australia. That model explained the high Denisovan ancestry in parts of Oceania, but it became less convincing as more genomes were studied. Different present-day populations carry Denisovan fragments, with different degrees of similarity to the Altai Denisovan genome. Some archaic segments appear to derive from a source relatively close to the Siberian branch. Others point to Denisovan populations that had been separated from the Altai lineage for a very long time. Recent work on near-Oceanian genomes strengthens the conclusion that at least 3 Denisovan-related populations contributed ancestry to the region's forebears. These contributions were not identical in age, genomic distribution, or evolutionary relationship. They suggest repeated contact, perhaps occurring at different stages as Homo sapiens moved through continental Asia, the Sunda region, and the island chains farther east. The precise number of encounters remains model-dependent. 3 inferred sources do not necessarily mean exactly 3 meetings. One donor population could have contributed ancestry repeatedly. Several related groups might be combined statistically into one source. Later mixing among Homo sapiens populations could redistribute archaic segments and make separate episodes appear closer together. What survives in the records suggests multiple contacts, but the original social and geographic pattern is more complicated than any single number. One encounter may have involved a northern or eastern Denisovan population with relatively close ties to the Altai lineage. Another may have occurred farther south with a deeply diverged group adapted to warmer environments. A third could represent an island or coastal population that had remained isolated for many thousands of generations. These possibilities fit the genetic evidence, but they are not yet tied securely to named archaeological sites. The movement of Homo sapiens through Asia was itself layered. Fossils and ancient genomes reveal early populations that did not contribute equally to later people. Some lineages expanded and then declined. Others mixed with newcomers and became difficult to distinguish. The roughly 40,000-year-old Tianyuan individual from northern China carried Denisovan ancestry, showing that archaic contributions were already present in early East Asians. Yet later East Asian groups did not inherit all archaic segments in the same proportions, while ancestry associated with the Jomon appears to reflect limited direct Denisovan contact. This pattern creates a map of near meetings and missed encounters. One Homo sapiens population could travel through territory where Denisovan-related groups were still present. Another could arrive after those communities had moved or declined. A coastline, mountain range, or ecological boundary might separate populations that never exchanged genes despite living within the same broad region. Timing mattered as much as geography. Population density was probably low. The meetings preserved in DNA may have involved relatively few people, but their descendants later became numerous. A small amount of admixture can leave a large modern signal if the mixed population expands while others disappear. The percentage of Denisovan ancestry today therefore does not measure how common Denisovans were or how often meetings occurred across the entire continent. It records the reproductive success of particular descendant populations. The genome cannot reveal how the participants understood one another. It cannot tell whether contact occurred through the joining of communities, movement of individuals, long-term coexistence, or brief episodes followed by separation. It shows only that some encounters produced descendants whose lineages continued. The people involved were genetically distinct, but they were not separated by an absolute reproductive barrier. Cultural contact may have accompanied biological mixture. Migrating Homo sapiens entered landscapes already known to local populations. Denisovan-related communities possessed knowledge of prey, water, seasonal plants, dangerous terrain, high-altitude conditions, and regional pathogens. Information could pass through observation or social interaction even when genes did not. Yet archaeology rarely allows a tool-making method or survival strategy to be assigned confidently to one population and shown moving into another. Similar technologies can arise independently, and artifacts do not carry genomes. The biological legacy is easier to trace. Denisovan-derived variants entered modern human populations and were then tested by new environments. Some disappeared quickly, others persisted neutrally. A limited number increased in frequency because they influenced traits useful under local conditions. The high-altitude EPAS1 haplotype is the most famous case, but immune-related archaic variants in populations of Oceania may represent another consequence of encounters with people who had lived in Asian environments far longer than the arriving Homo sapiens groups. More than 3,000 Denisovan-related variants identified in near-Oceanian genomes may influence gene activity. Many are associated with immune pathways, including responses involving interferon signaling. This does not mean all were advantageous or that each can be connected to a specific ancient disease. Functional effects are complex, and modern environments differ from Pleistocene ones. Still, the pattern suggests that introgression sometimes gave expanding populations access to variants already shaped within local ecological conditions. The exchange also carried costs. Archaic ancestry is reduced in many functionally important regions of modern genomes. Natural selection appears to have removed variants that interacted poorly with Homo sapiens biology. Reproductive genes and regulatory systems can be especially sensitive to combinations formed after long population separation. The surviving Denisovan legacy is therefore the remainder after tens of thousands of years of filtering, not the full genetic contribution made during the original encounters. Each modern population preserves a different selection of fragments. Two people can share Denisovan ancestry without carrying exactly the same Denisovan segments. Recombination repeatedly breaks chromosomes, while chance determines which pieces pass onward. Over generations, the archaic genome becomes scattered across many descendants. No living person contains a complete Denisovan ancestor within them. Collectively, however, populations preserve a larger catalogue than any single individual. This collective archive now reveals Denisovan diversity beyond the fossils. The Altai genome once seemed to represent the population as a whole. Modern human genomes show that it represents only one branch. The people encountered by ancestors of near-Oceanians were separated enough from one another that their contributions remain distinguishable after tens of thousands of years. In a sense, Homo sapiens genomes have preserved samples from Denisovan populations whose own bones have not yielded DNA. But this archive is distorted. Selection favors some segments and removes others. Population bottlenecks exaggerate certain contributions. Later migrations blend ancestry acquired in different places. Researchers must reconstruct the original events from a record repeatedly edited by biology and history. Scholars still debate the dates and locations of particular admixture episodes, and no model captures every movement across Asia. What is increasingly clear is that the arrival of Homo sapiens did not trigger one simple replacement. In some regions, modern humans may have lived near Denisovan-related groups for long periods. Elsewhere, contact may have been brief. Some Denisovan populations disappeared without leaving detectable descendants. Others entered expanding Homo sapiens communities so thoroughly that their independent history ended while portions of their ancestry continued. This is one way a population can vanish without being completely erased. Its communities cease to exist. Its language falls silent, and its territory passes to others. Yet fragments of its biology travel onward in people who no longer know where those fragments came from. The Denisovan populations encountered across Asia became invisible in precisely this way. Their differing genetic contributions also expose a deeper problem. If 3 Denisovan sources can be distinguished in near Oceania, then the name Denisovan may conceal more diversity than it reveals. The next layer of the mystery is not simply where Denisovans lived, but whether science is dealing with one population, several subspecies, or an entire family of deeply separated human groups gathered under a name created from a single Siberian cave. A name created from one Siberian cave now stretches across nearly an entire continent. That alone should make it suspect as a description of one uniform people. The Altai Denisovan genome was the first clear signal, so every later fragment of related ancestry was initially measured against it. But as more modern and ancient genomes were studied, the pattern became increasingly difficult to compress into a single population. The people called Denisovans were related to one another, yet some of their branches has been separated for so long that they may have differed in appearance, adaptation, and culture as profoundly as widely scattered human populations can differ today. The first strong genetic warning came from Papua. Researchers found that Papuan genomes did not preserve ancestry from only one Denisovan source. At least 2 deeply divergent Denisovan-related lineages appeared necessary to explain the pattern, and one was substantially more distant from the Altai genome than expected. This meant that the group encountered by ancestors of Papuans could not be represented adequately by the individual from Denisova Cave, somewhere farther south or east. Another Denisovan population had followed its own evolutionary path for an immense span of time. More recent genomic work in near Oceania has enlarged that picture again. The ancestry of communities across the region is best explained by contact with at least 3 populations related to Denisovans. The exact number may change as sampling improves and models become more sophisticated, but the direction of the evidence is clear. Denisovan ancestry is internally structured. It entered modern human populations through several sources, rather than flowing from one genetically homogeneous group. These sources are reconstructed from differences among inherited segments. Some fragments carried by living people are relatively close to the Altai Denisovan sequence. Others branch much deeper. Their mutations, lengths, frequencies, and relationships across the genome suggest donors whose ancestors had separated from one another long before the meetings with Homo sapiens. Later recombination shattered the original contributions, but enough of their distinct histories survive for geneticists to tell that they did not all come from the same source. This does not automatically mean that 3 Denisovan species existed. Species boundaries are difficult to establish even when complete skeletons and genomes are available. The inferred groups could have been regional populations within a broad Denisovan lineage, maintaining occasional contact while remaining largely separated by distance and ecology. Mountain chains, deserts, forests, coastlines, and changing sea levels could isolate communities for thousands of generations without creating an absolute reproductive barrier. The Altai branch may represent only a northern population adapted to cold and strong seasonality. Denisovan-related people on the Tibetan Plateau faced low oxygen and limited resources. The Harbin individual belonged to the river landscapes of northeastern China. Penghu lived somewhere near the subtropical plains now submerged beneath the Taiwan Strait. Farther south, the donors of substantial Oceanian ancestry may have occupied environments from monsoon forests to coastal lowlands and islands. Calling all of them Denisovan is genetically useful, but it should not erase their likely differences. Their bodies may have varied as well. The robust Siahe and Penghu jaws share certain broad features, while the Harbin skull provides a much fuller but geographically limited portrait. None can be treated as a universal Denisovan form. Long-separated populations could evolve different cranial shapes, tooth sizes, limb proportions, and physiological traits. Interbreeding with neighbouring groups would add further variation. A southern Denisovan population might not have looked exactly like the northern individual reconstructed from Harbin, just as the communities carrying distinct Denisovan ancestries need not have shared one appearance. Their cultures were almost certainly regional. A tool tradition suited to the Altai cannot be assumed to extend unchanged into tropical Southeast Asia. Stone itself may have played different roles depending on available raw material. In bamboo-rich environments, much daily technology may have been organic and therefore lost. Plateau populations required insulation, fuel management, and knowledge of high-altitude animals. Coastal communities may have relied on rivers, wetlands, shellfish, or shoreline resources. The archaeological record is too sparse to assign these lifeways securely, but ecological variation makes a single Denisovan culture implausible. Population size may have intensified the separation. Genetic evidence suggests that some Denisovan groups remained small for long periods. Sparse populations distributed over vast territories can diverge rapidly through isolation, local selection, and chance. Occasional meetings may reconnect them, but not enough to eliminate regional differences. The result is a population complex related groups sharing deep ancestry, while following distinct histories. Modern humans did not encounter this complex all at once. Different Homo sapiens populations travelled through different parts of Asia at different times. One group may have met a northern Denisovan branch, another encountered a southern population much later. A third acquired ancestry indirectly through mixture with an already admixed Homo sapiens community. Later migrations then brought these contributions together, making the final pattern appear within the same modern populations. This layered process helps explain why Denisovan ancestry is unevenly distributed. People in parts of Oceania preserve relatively high levels, while many mainland Asian populations carry smaller amounts. Some early East Asian individuals held Denisovan segments that later became rare or disappeared. Other populations seem to have experienced little direct contact. There was no single continental wave in which every modern human group met the same archaic population. The ancient genomes sampled so far are also biased. Cold northern sites preserve DNA far better than southern ones. So the best Denisovan genome comes from a region that may have been peripheral to much of Denisovan diversity. The populations that contributed most strongly to Oceanian ancestry remain genetically visible only through their modern descendants. Their own genomes have not been recovered. This creates an inverted archive. The most complete ancient sample may represent one branch, while the most influential southern branches are known only from the pieces they left in another population. Fossils may eventually correct that imbalance. Harbin demonstrates that molecular evidence can attach a nearly complete skull to Denisovan ancestry. Penghu shows that tooth proteins can identify a fossil even when DNA has vanished. Dental calculus, enamel proteins, sedimentary DNA, and improved screening methods may reveal Denisovan connections among remains already stored in museums. Some fossils now assigned to broad Middle Pleistocene categories could belong to different Denisovan branches, although each claim will require independent evidence. There is also a risk of allowing the Denisovan label to become too broad. Not every unexplained Asian fossil or archaic DNA segment should be placed within it. Asia contained Homo erectus, island populations with distinctive anatomies, Neanderthals in its western and northern reaches, and perhaps other groups that remain unknown. Some genetic signals may come from lineages outside the Denisovan family entirely. A convenient label can become another form of blindness if it absorbs every mystery. This safer interpretation is that Denisovans provide the clearest example of a wider phenomenon. A population first recognized as one genetic ghost turned out to contain several ghosts within it. The genome from the Altai revealed the lineage, but modern DNA exposed its internal diversity. Fossils are only beginning to attach bodies to those branches. This changes the central question. It is no longer enough to ask what Denisovans looked like or where they lived. One must ask which Denisovans. The northern cave population, the high-altitude groups, the northeastern Chinese branch, the subtropical people near Taiwan, and the deeply divergent donors represented in Oceania may belong to one broad ancestry without sharing one history. The term Ancestor X becomes more accurate when treated as plural. Some invisible ancestors are not wholly unknown lineages but unsampled branches within a lineage already recognized. Others may be more ancient and more difficult to place. Denisovan diversity shows how easily a single recovered genome can create the illusion of unity. It also prepares the way for a deeper possibility beneath the Denisovan branches themselves, and perhaps beneath the ancestry of all modern humans. Genetic models detect contributions from populations separated by far greater depths of time. Beneath the Denisovan branches lies a deeper possibility. The genetic ghosts in living humans may not all belong to populations as recognizable as Neanderthals and Denisovans. Some models point toward lineages that separated from the ancestors of known humans so deeply in time that even the word archaic begins to feel inadequate. These populations have no confirmed skull, no secure species name, and no ancient genome. They appear only because certain parts of our ancestry seem to have followed a path that ordinary branching models cannot easily explain. One of the most provocative recent reconstructions concerns the ancestry shared by all living humans. Using a structured genetic model, researchers proposed that two ancestral populations separated roughly 1.5 million years ago, remained apart for an immense span, and came together again around 300,000 years ago, near the broad period in which Homo sapiens was emerging. In the model, one population contributed most of the ancestry at that reunion, while the other contributed roughly 1/5. The smaller contribution has often become the headline, but the number requires careful handling. It represents an estimated proportion at a modelled admixture event. It does not mean that every person alive today still carries an intact 20% inherited from one mysterious species. Recombination, natural selection, and later migrations have repeatedly broken, removed, preserved, and redistributed that ancestry. The 20% figure is therefore not a simple measurement taken directly from a modern genome. It is the outcome of a model designed to explain patterns of shared ancestry, divergence, and selection. The identity of the smaller source is unknown. Homo erectus is an obvious candidate in popular retellings because populations assigned to that broad species existed across Africa and Eurasia during the relevant period. Homo heidelbergensis and other Middle Pleistocene forms are also invoked, yet no fossil can currently be connected securely to the modelled contribution. Without DNA or a uniquely diagnostic molecular signature, attaching the signal to a named species would be speculation. The mystery deepens because similar patterns appear elsewhere in the human family. Models of Neanderthal and Denisovan ancestry suggest that their common ancestors may have interbred with a much more deeply divergent Eurasian population more than 700,000 years ago. This inferred group is often called superarchaic. The term sounds formal, but it is not. It describes genetic distance, a population whose lineage may have separated from the ancestors of later humans close to 2 million years ago. Again, Homo erectus becomes a tempting possibility. Early Homo populations had already expanded beyond Africa, leaving fossils from the Caucasus, East Asia, and Southeast Asia. A long-established Eurasian population could have met the ancestors of Neanderthals and Denisovans when those newcomers entered regions occupied by older humans. The geography and timing are plausible. The genetic identification is not. Superarchaic may correspond to Homo erectus, to several related populations, or to a lineage not represented clearly in the known fossil record. Some newer preprint models propose multiple episodes of introgression from deeply divergent Homo lineages into modern human and Denisovan ancestry. They may refine the network, but have not completed peer review and depend strongly on assumptions about mutation rates, generation times, population sizes, and allowed histories. This is the central problem of Ancestor X. Geneticists do not excavate the unknown population directly. They infer it from patterns that remain after almost every original detail has been erased. If a segment of DNA branches unusually deeply, one explanation is that it entered through interbreeding with a long-separated population. Another is that the ancestors of Homo sapiens were themselves divided into regional groups for long periods, maintaining only limited contact. Both processes can produce old genetic lineages within a later population. Africa is where this ambiguity becomes especially important. Studies of West African genomes have reported signals consistent with introgression from an unknown archaic population that diverged before the separation of modern humans and Neanderthals. Some estimated contributions were substantial, yet ancient DNA from the likely donor has never been recovered, and later work demonstrated that persistent population structure can imitate some of the same signals. Imagine several ancestral African populations connected by occasional migration rather than one freely mixing population. A lineage preserved in one region could become highly divergent while still belonging to the wider ancestry from which Homo sapiens eventually formed. When those populations later mixed more extensively, the old lineage would appear as a deep block of ancestry. A model assuming one ancestral population might interpret it as DNA introduced by a separate archaic species, even though no such later interbreeding occurred. The weakly structured stem model offers precisely this alternative. Instead of Homo sapiens emerging from one isolated source, it proposes several ancestral African populations that remained partly connected across hundreds of thousands of years. Modern humanity then formed through their repeated interaction. Under this view, some ghosts in the genome are not outsiders entering our lineage. They are long-separated parts of the lineage itself. Ancient genomes from southern Africa add weight to the need for caution. Deep structure can reflect long isolation among Homo sapiens populations, archaic admixture, or both. And the evidence does not always separate them cleanly. An ancient lineage unknown before a genome is sequenced is not automatically an unknown species. The Green Sahara genomes from Takakori revealed previously unrecognized North African ancestry, but that ancestry belongs within Homo sapiens rather than demonstrating a separate archaic human. Names can therefore mislead. A ghost population is not necessarily a ghost species. Unknown ancestry is not necessarily archaic ancestry. A superarchaic signal is not a fossil classification. Even the word population may describe a statistical source, rather than one clearly bounded community. Researchers use these terms because the genetic pattern requires some historical explanation. not because the people behind it have been fully identified. New tools attempt to improve that reconstruction. Methods based on genome-wide genealogical trees can search for archaic segments without relying on a known donor genome. Trace identifies regions whose local family trees branch in ways expected after introgression. Ghostbuster attempts to reconstruct mixtures involving unsampled populations. These approaches can detect known Neanderthal and Denisovan signals, and also report deeper candidate segments from unidentified sources. But methods must be tested against histories in which no ghost admixture occurred. Simulations show that when ancestral population structure is ignored, some techniques infer interbreeding that was never built into the simulated past. This does not invalidate the well-supported evidence for Neanderthal or Denisovan ancestry, because their genomes provide direct reference points. It does mean that claims involving an unsampled Ancestor X demand greater restraint. The most honest conclusion is also intriguing. Human ancestry almost certainly contains more structure and more episodes of contact than the old tree allowed. Some involved known groups. Others may involve populations represented by fossils but not yet genetically identified. Still, others may be statistical shadows created by the long separation and reunion of ancestral Homo communities. No single explanation fully accounts for all the evidence. Ancestor X is therefore not waiting as one missing skull that will solve the problem. There may be several ancestors, several reunions, and several meanings hidden beneath the same genetic pattern. A future genome could reveal that one ghost belonged to a known fossil population. Another may dissolve into a better model of structured ancestry. A third may remain permanently ambiguous because no recoverable molecule survives. The deeper the analysis moves, the less human evolution resembles a line of named species. It becomes a landscape of populations separating, exchanging genes, and merging until their identities are no longer recoverable as distinct groups. The mystery is not only who Ancestor X was, it is whether the past itself ever contained the clean boundaries required for that question to have one answer. Disappearance rarely happens at one moment. A population does not simply step out of history because a colder winter arrives, a rival group appears, or one migration crosses a frontier. More often, disappearance is the final name given to a long process. Communities shrink, territories fragment, birth rates fall, local groups lose contact, and surviving individuals enter other populations. By the time archaeology notices the absence, the social world has already been transformed beyond recognition. For Denisovan-related populations, there is no single extinction layer, no continent-wide catastrophe marks their end, and no final settlement can be identified as the last place where Denisovans lived as a distinct community. Their fossils span separated regions and periods, while their genetic legacy shows that some groups survived long enough to meet Homo sapiens repeatedly. The end must therefore have unfolded unevenly across Asia. Climate instability offers one part of the explanation. The late Pleistocene brought repeated shifts in temperature, rainfall, vegetation, and sea level. Forests became grasslands, river systems changed course, and glacial cycles altered migration routes. Populations adapted to narrow ecological zones could be vulnerable when familiar prey declined or local resources moved beyond reach. Even adaptable groups faced limits if several changes occurred quickly, or if small communities became trapped in shrinking habitats. The Altai region illustrates this instability. Climatic phases reorganized the valley around Denisova Cave, changing which animals and human populations could enter. A region that served as a refuge during one climatic phase could become difficult or inaccessible during another. Repeated local abandonment would reduce continuity even without causing immediate extinction. High-altitude groups face different pressures. On the Tibetan Plateau, survival depended on seasonal resources, reliable fuel, and access to animals adapted to thin air. Coastal and tropical populations faced monsoon changes, forest contraction, flooding, and the loss of a lowland territory as sea levels rose. Some landscapes that may have supported southern Denisovans now lie beneath the sea, erasing both habitat and evidence. Small population size may have been even more dangerous than climate alone. Genetic data suggest that Denisovan groups experienced long periods of low effective population size and deep regional separation. Small populations are vulnerable to chance, a few poor seasons, disease outbreaks, or failed movements can reduce numbers sharply. Limited access to unrelated partners can increase inbreeding and lower resilience. Knowledge can also disappear when too few people remain to maintain it. This does not require a dramatic collapse. A community can survive for generations while becoming progressively more fragile. One valley is abandoned. Another group fails to reconnect with distant relatives. A regional tradition disappears because only a few families remain. Over thousands of years, a continent-wide population complex can break into isolated remnants with little ability to recover after local losses. The arrival and expansion of Homo sapiens added another pressure, but not one that should be reduced to a simple invasion story. Modern humans did not necessarily defeat Denisovans in direct conflict across Asia. Evidence for such widespread confrontation does not exist. Competition could operate more quietly through population size, mobility, social networks, and the ability to recover after setbacks. Larger and better connected Homo sapiens populations may have occupied key territories and expanded into ecological gaps that smaller archaic groups also depended upon. Technology may have mattered, but claims of universal modern human superiority are difficult to sustain. Denisovans survived in demanding environments and possessed effective local knowledge. Homo sapiens' advantages may instead have been demographic and organizational wider exchange networks, greater movement of materials and information, or more frequent contact between communities. Even modest differences in population growth can transform a landscape over many generations. Disease is another possibility, though direct evidence is scarce. Populations separated for long periods can develop different immune histories, and renewed contact may expose each group to unfamiliar pathogens. Denisovan-derived immune variants in modern people show that archaic populations carried important adaptations, but they do not reveal whether epidemics contributed to Denisovan decline. The idea remains plausible, not proven. Interbreeding complicates the meaning of disappearance most of all. Some Denisovan communities may not have been destroyed or displaced. They may have been absorbed. When a small population mixes with a much larger one, its distinct identity can vanish even while some ancestry survives. Children inherit both lineages, and later generations become increasingly integrated into the larger population. Eventually, no separate Denisovan community remains, but Denisovan DNA continues within Homo sapiens' descendants. This process may explain part of the uneven genetic legacy. Some Denisovan groups contributed substantial ancestry to expanding modern populations. Others left little or none. A community can be biologically absorbed only if contact occurs and descendants survive. Isolated groups may disappear without leaving detectable traces. Living genomes therefore preserve the populations that met reproductively successful Homo sapiens groups. not every Denisovan population that existed. Natural selection then edited the inheritance. Some archaic segments were removed because they reduced reproductive success or interacted poorly with modern human biology. Others persisted by chance, and a few became beneficial. The surviving fragments are not a complete memorial. They are a filtered residue shaped by demographic expansion and selection. Regional diversity means that the end probably happened at different times. Northern Denisovans could have disappeared long before some southern populations. High-altitude communities may have persisted after lowland relatives were absorbed. A deeply diverged branch in Southeast Asia may have encountered Homo sapiens later than Altai populations did. The last Denisovan community may never be identified because it lived where fossils and DNA rarely survive. There is also the possibility that the label itself conceals different endings. If Denisovans were a collection of related populations rather than one homogeneous group, some branches may have merged with Neanderthals, others with Homo sapiens, and still others with unknown Asian populations. The disappearance of the Denisovans could be shorthand for regional transformations, separated by tens of thousands of years. Scholars still debate the balance among climate, demography, competition, disease, and assimilation. No single explanation fully accounts for the evidence. Climate change alone cannot explain why Homo sapiens expanded through some of the same environments. Competition alone cannot explain repeated interbreeding. Assimilation alone cannot account for branches that left no detectable ancestry. The most realistic picture combines several processes. Imagine communities spread thinly across Asia. Some occupy cold valleys, others plateau caves, river plains, forests, or coastlines. Their populations are small, their networks uneven, and their contact intermittent. Climate repeatedly alters the routes between them. Modern humans enter from several directions, sometimes competing, sometimes exchanging knowledge, sometimes forming families. One Denisovan community is absorbed. Another moves and loses contact with its relatives. Another declines after ecological change. A final group survives for generations in a region that leaves no recoverable DNA. Nothing in this process requires a last day. The disappearance occurs when the network no longer reproduces itself as a distinct human world. Tools continue to be made, fires continue to burn, and people continue to live. But identities and ancestries blend. The population name used by future science has no meaning to those experiencing the change. By the end, Denisovans are absent as a recognizable later population, yet not fully gone. Their high-altitude adaptation remains in Tibetan genomes. Immune-related variants persist in parts of Oceania. Their ancestry survives in scattered fragments across Asia and beyond. Harbin, Siahe, Penghu, and Denisova Cave recover pieces of their bodies, while modern genomes preserve pieces of their inheritance. This is not survival in the cultural sense. Their languages, traditions, and social memories did not continue in a form that can be identified. But extinction is no longer a clean word either. A people can disappear as a population and remain present as inheritance. Denisovan history ends not with a single silence, but with absorption into a larger human story whose final shape they helped create. The larger human story still carries them, not as complete ancestors preserved intact inside any living person, and not as a hidden species waiting to reawaken, but as scattered pieces of inheritance passed through thousands of generations. Denisovan-related DNA survives unevenly across Asia and Oceania, shaped by the routes modern humans travelled, the populations they encountered, and the environments their descendants later entered. The pattern is not a genetic monument placed deliberately in the future. It is the accidental survival of fragments from meetings that no one at the time could have known would matter tens of thousands of years later. In some populations of near Oceania, Denisovan ancestry remains more abundant than anywhere else known. Yet even there, it does not come from one uniform source. The inherited segments point toward at least 3 Denisovan-related populations, each carrying a separate history before contact with Homo sapiens. Some fragments resemble the Altai-Denisovan genome more closely. Others derive from branches that had already been isolated for long periods. Together, they preserve a faint genetic map of populations for which almost no direct fossil evidence has yet been recovered. This map is incomplete and distorted, but it contains functional traces. Thousands of Denisovan-derived variants identified in near-Oceanian genomes may influence the activity of modern genes. Many occur within biological pathways connected to immunity, including signaling involved in responses to infection. The ancient populations who lived across Asia encountered local pathogens long before arriving Homo sapiens groups did. Some of their inherited variants may therefore have offered useful responses when carried into newly mixed populations. The effect was not automatically beneficial. Immune systems balance protection against the risk of excessive inflammation or mistaken attack on the body itself. A variant useful under one pathogene environment can become neutral or harmful under another. Modern studies can identify associations and test changes in gene activity, but they cannot fully reconstruct the disease landscapes in which natural selection operated. What survives in the record suggests that certain archaic variants mattered, while the exact advantage they once provided often remains uncertain. The same caution applies to Denisovan influences on metabolism, body regulation, and other traits. Archaic ancestry can interact with modern genomes in complicated ways. A segment may affect several biological processes at once. Its impact may depend on diet, altitude, climate, or ancestry elsewhere in the genome. The idea that Denisovans handed modern humans a series of ready-made adaptations is too simple. Introgression created variation, natural selection, chance, and later demographic history decided which pieces endured. The EPIS 1-related high-altitude adaptation remains the clearest example. A Denisovan-related sequence entered the ancestry of Homo sapiens and later became common among Tibetan populations living under chronic low-oxygen conditions. The variant helped shape a physiological response that avoided some of the costs associated with excessive red blood cell production. It did not make altitude effortless, and it functioned alongside many other genetic and cultural adaptations. Yet its survival forms a direct biological connection between present-day plateau communities and an archaic population that had occupied high elevations long before. This inherited solution reveals the strange logic of extinction. Denisovan communities disappeared, but one part of their response to thin air continued in another population. Their adaptation outlived the social world in which it developed. A biological trait crossed a boundary that language, tradition, and memory did not. Elsewhere, Denisovan ancestry survives in smaller amounts across East, South, and Southeast Asia. The distribution reflects ancient encounters followed by later mixing. Some populations received ancestry directly from Denisovan-related groups. Others inherited it indirectly from neighboring Homo sapiens populations that had already mixed with them. Migrations spread fragments beyond the places where the original meetings occurred, while population replacements removed them from regions where they may once have been more common. Ancient genomes reveal this movement through time. Early East Asians sometimes carried Denisovan ancestry in patterns unlike those of later populations. Some segments disappeared through drift or selection. Others were diluted when new populations arrived. The genetic landscape of the present is therefore not a simple photograph of the late Pleistocene. It is the latest frame in a long sequence of migrations, bottlenecks, expansions, and reunions. Even the word legacy can mislead if it suggests intention or cultural continuity. Denisovans did not knowingly leave genes to future populations. The inheritance came through ordinary reproduction among people living in uncertain circumstances. Nor does carrying Denisovan DNA create a direct cultural identity with Denisovans. A person may inherit an archaic segment without inheriting any memory of the population from which it came. Genes travel farther than stories. Still, the fragments have scientific power. They allow researchers to infer populations whose own genomes have never been recovered. By comparing Denisovan-derived segments across living people, scientists can estimate how deeply the donor groups had diverged, when admixture may have occurred, and which parts of the archaic contribution were preserved or removed. Modern genomes become archaeological sites distributed across millions of bodies. These biological archives must be approached with care. The individuals whose DNA is studied belong to living communities with their own histories, rights, and relationships to research. Near-Oceanian and indigenous populations are not containers for ancient ancestry. Their genomes reflect complex modern identities and demographic histories extending far beyond Denisovan admixture. Responsible research depends on collaboration, informed participation, and a refusal to reduce present-day peoples to prehistoric evidence. The same care is needed when discussing ancestry percentages. No population is more archaic, less evolved, or closer to an ancient human type because it carries a higher proportion of Denisovan DNA. Every living person belongs equally to Homo sapiens, and every population has evolved continuously through the present. Archaic ancestry records historical contact, not a hierarchy of humanity. Denisovan inheritance also places the broader Ancestor X problem in perspective. Here is a ghost population that moved from statistical signal toward physical recognition. First came unexpected DNA from a tiny finger bone, then came additional genomes, teeth, jaws, proteins, sediments, and eventually a likely face in the Harbin cranium. The ghost was not invented by the model. Molecular evidence repeatedly connected it to real bodies across Asia, but even this successful deghosting remains unfinished. The donors of much Oceanian Denisovan ancestry have not been identified among known fossils. The number of Denisovan branches remains uncertain. Their southern range is largely inferred. Their languages, cultural systems, and final communities remain beyond recovery. Science has transformed Denisovans from an unknown lineage into a population complex, yet the clearer picture has produced more questions than it resolved. Other genetic ghosts may follow the same path. A fossil already resting in a museum drawer could belong to one of the unsampled lineages inferred from modern DNA. Tooth enamel may preserve identifying proteins. Dental calculus may contain mitochondrial fragments after bone has failed. Cave sediments may reveal populations that left no recognizable skeleton. New methods may recover family trees from genomes without requiring a donor reference. Some ghosts, however, may disappear under better analysis. A signal interpreted today as archaic admixture could prove to reflect ancient population structure. A supposed unknown species may turn out to be a long-isolated branch of early Homo sapiens. A model proposing one dramatic reunion may be replaced by centuries of intermittent contact among several populations. Scientific progress does not only reveal hidden ancestors, it also removes ghosts created by assumptions. This is why Ancestor X must remain a question rather than a name. It represents the space between a genetic pattern and a human population that can be demonstrated. Sometimes that space contains Denisovans. Sometimes it may contain a deeply divergent African population, an early branch of Homo sapiens, or a Eurasian lineage related to Homo erectus. Sometimes it contains several histories layered together. The deepest model suggests that all living humans may descend from ancestral populations that separated more than 1 million years ago and later rejoined. If this reconstruction is broadly correct, then mixture is not a minor event added to an otherwise simple origin. It is part of the foundation of modern humanity. The population that contributed the smaller modelled share did not survive under a recognizable name, yet its involvement may reach across every living human lineage. No fossil has been securely identified as that population. The 773,000-year-old remains from Morocco lie near a crucial region and period in the emergence of the Homo sapiens lineage, but they do not provide the DNA needed to link them to the modelled ancestry. Other fossils across Africa may occupy related positions. Their anatomy preserves mosaics of older and more derived traits, exactly what might be expected from a world of structured populations and repeated contact. But resemblance is not identification. The search will continue through genomes, proteins, sediments, and bones. Each new result will need to survive alternative explanations. A deep branch must be tested against population structure. An apparent admixture event must be reproduced by independent methods. A molecular fossil identification must be checked against contamination and preservation. Preprints will need peer review, and dramatic percentages will need to be translated into what the models actually estimate. The reward for that caution is a richer history. The human past is not diminished when certainty is limited. It becomes more honest. We are the descendants of populations whose boundaries shifted, whose descendants merged, and whose names are lost because names rarely survive deep time. Some left skulls, some left tools, some left only a pattern of mutations moving quietly through later generations. Denisovans are the clearest reminder that absence from the fossil record is not absence from history. For most of their scientific existence, they were almost invisible. Yet their descendants in the genetic sense were already living across the world, carrying evidence before anyone knew how to recognize it. The unknown human had never truly vanished. Science had simply not learned where to look. And now, as the search reaches deeper into the genome, the boundary between the lost and the living becomes softer. A cave in Siberia, a jaw from a plateau, a skull from northeastern China, and a sequence carried across islands all belong to the same unfinished story. The people themselves are gone, their communities dissolved into time, but the traces remain, not as complete portraits, only as fragments asking to be placed beside one another. Perhaps that is the final shape of deep human history, not a gallery of perfect ancestors standing in order, but a dark landscape crossed by many small fires. Some burn brightly enough to leave cities, monuments, and written names. Others fade before history begins. Wind passes over their camps. Rivers alter their course. Caves fill slowly with sediment. Seas rise over the plains where they once walked. For a long time, nothing seems to remain. Then a tooth is lifted from the ground. A fragment of bone yields a damaged sequence. Mineralized plaque preserves a line of mitochondrial inheritance. A living genome carries a segment whose branch reaches farther back than expected. The fire is gone, but one ember has travelled onward. It passes without memory from parent to child, through migrations and changing languages, through winters, coastlines, and generations too numerous to count. It becomes smaller. It is broken and joined to other inheritances. Sometimes selection removes it. Sometimes it remains because it helps a body breathe, respond to infection, or simply survive by chance. No one carrying it can hear the vanished voices within it. DNA preserves relationship, not recollection. It cannot restore the name of a Denisovan community or tell us what a person saw while standing above the Anui River. It cannot reveal the stories spoken inside Oceania Cave while cold air moved across the plateau. Those worlds are beyond return, yet their silence is not emptiness. It is the silence of a past that still touches the present without explaining itself. Every newly recovered fragment reminds us that humanity was once plural, that several kinds of humans shared the earth, and that their separations were never as complete as later categories suggested. Far back in time, populations moved toward one another across changing landscapes. For a moment, their histories met. Then the people disappeared, while part of the meeting continued, and somewhere within the quiet architecture of a living cell, a fragment from that encounter still waits small, altered, almost anonymous, carrying the memory of no single ancestor, but the lingering shadow of an entire human world.